Association. We meet when we're meeting officially, we meet in Toledo. Of course, like everybody else, we're doing the Zoom thing right now with the help of Jamie Walters, which I really appreciate. One of the things I did put in a newsletter this morning that the research girl, Harper, who's working with Ohio State on what bees are working, what kind of flowers are working. Her and I put the hive scales out that the Maumee Valley Beekeepers bought, or put some money up to buy, and I don't know if Dwight Wells is on here. We thank Dwight too. He furnished quite a few. If you don't know, Ohio State is pretty much locked down research money for the summer because of the COVID thing. So she was kind of caught between a rock and a hard place, and the Maumee Valley Beekeepers Club stepped up and helped her out, and I'm helping her out. We went to nine of my apiaries, put some scales on, and I'll be collecting honey for her also. So it's an ongoing thing. She's in fact already talking that this could be more than a one-year project. So she wants to find out where our bees are going and how much farming impacts it. I'm in the high agriculture impact areas. So, but our club has about 75 paid members these days, and we've been around for a while. We'd be glad to have anybody that wants to join. Our dues are $10 a year. So we try to get some guest speakers around for most of the meetings. I get tired of talking to everybody, so I try to find somebody else to do the talking. So glad to be here, and I'm glad this worked out, and I hope some more people come on. So welcome, everyone.
I'm Jamie Walters, the president of Black Swamp Beekeepers, and at this time, the same way with COVID. So everything's on a Zoom call or either a Zoom presentation. With the membership, you're welcome to join our club. We're the second Tuesday of every month. Dues are $20 per individual or $25 per family per year. This year, with everything going on, we've stayed pretty active out of the community apiary with visitations and things going on. We still have the beginner internship that's going on from February. It started this year in March, and we're going to go all the way to November. We have our potluck party. Chris Copes has reserved that back, so we're going to kind of play that by ear. We're still going to have our potluck party at the end. Other than that, it's mostly education online, as everybody's known about this. And we kind of jumped in with OSBA, and I've been doing a lot of Zoom calls on that and trying to keep the education out there, because just as the beginning classes got in for the beginning beekeepers, then all of a sudden COVID hit, and now all these beekeepers are left out in the kind of the area of nothing. So we're trying to get some stuff out to them. I want to thank everybody for jumping on the call tonight and spending your afternoons. And like I talked earlier, you don't have to stay here all day, but there's the lineup that's on your screen now, and you're welcome to jump in and out. I do ask for you to keep your mics on quiet, that you're not going to interrupt the call. These will be recorded, and then we're going to decide, we're going to get permission from all the speakers, and then we'll see where we can do with that from there. Mr. Russell Bowes.
Good afternoon, everybody. My name is Russ Bowes. I'm with the North Central Ohio Beekeepers Association. We just started up in last year, 2019. We've got between 12 and 15 members right now. Memberships are $10 a piece. We meet in Upper Sandusky. We were starting to meet in Upper Sandusky, but because of COVID, of course, we're not meeting in face-to-face. Jamie is helping us with the Zoom meetings. Appreciate that, Jamie. Our meetings are the second Wednesday of each month.
Jerry Hayes, you have the floor. Go ahead and share your screen.
Okay, let's see if this works, Jamie. Jamie gave me a lesson last night, and I'm not too bright, so let's just see what happens here.
Perfect job, Jerry.
Well, it's not over yet, Jamie, so.
And Mr. Hayes, take it away.
Well, thank you, Jamie. I certainly appreciate that. And I certainly appreciate the opportunity to be able to talk to whoever is out there. It looks like a beautiful day out there. And if you're out working your bees, I don't blame you a bit. But you do have a great lineup of speakers and topics and great leadership in your local organization and your state organization. And there again, thanks to Jamie for being technologically more advanced than most of us are. So appreciate that. And so going to spend a few minutes with you talking about nutrition. My goal is to confuse and probably upset you and some suppliers. So if I do that, I will have done my job. Nutrition. Everybody here probably had some form of breakfast, or you're going to be eating a sandwich while you're watching one of these. And nutrition is key to not only our health, but the health of the bees as well. And I'm trying to advance, Jamie, and it's not doing that. What am I doing wrong here?
You should just be able to click your mouse, click on your presentation.
Yeah, that's what I was thinking. And it's oh, there we go. Okay.
There you go.
Thank you, Jamie.
You're
I had
welcome.
to press it harder, apparently. All right, everybody's familiar with, hopefully, the food pyramid. What foods are necessary, good for us, because they give us the protein, the vitamins, and the minerals that allow us to be us, to allow us to grow, replace cells, and in this time of COVID, to support our immune systems so that we can fight off various diseases. And so, as you can see, we have herbs and grains and proteins and vegetables, and this mixture of foods for us is what keeps us healthy and active and have the energy to be beekeepers. But nutrition is different for us, for humans, at different parts of our lives. When we're young, you don't have a baby and feed it a peanut butter and jelly sandwich or garlic bread or what have you. Mother's milk at this point in time is the best nutrition for babies because their gastrointestinal system is designed to extract the nutrients from that easier than when they get a little bit older, where they can eat some solid or semi-solid foods as they grow and develop. And then, of course, there's sports nutrition. If you're in sports or riding a bike or out playing golf or, being a beekeeper all day long, that nutrition might be different than somebody who's spending their day in the library. And then, the concept is we have to eat properly in order to be our best and to do our best and to win whatever it is you want us to win. But there again, as we age, that nutrition changes also. Our gastrointestinal system changes. And that's a picture of me this morning eating my breakfast. And as you can see, it's a little bit different than, I might have had when I was 20 or 30 and having the fried eggs and the bacon and what have you, because our systems change. So we have to have different foods, perhaps prepared differently as we age and grow and what our activities are. And it's the same with animal nutrition. A baby animal, has something different to eat than a mature animal. Chickens and cows and the pigs and what have you, and farmers and what have you, and fish have to feed these animals diets that will allow them to grow and then be harvested as well. And then we don't think about this, but we know how to feed every animal in the zoo. So there's also zootrition. We know how to feed elephants and aardvarks and penguins and everything else so that they can grow and develop, and some zoos do, to have baby rhinoceroses and what have you. So diets and nutrition are well researched for many, many different kinds of animals, for people at different ages, so that we can bring those people and animals to their very best. But for supplemental nutrition for honeybees, we know a lot, but we haven't incorporated it yet. But we know that honeybee nutrition, and of course, the bees are out flying well today and finding flowers and collecting pollen, which is their protein, vitamin, and mineral source when it's turned into bee bread, in order to keep them healthy and to feed their baby sisters. There's a great paper out there from a few years ago even, called Fat Bees, Skinny Bees. And I would encourage each and every one of you to Google this up and get a copy. It's actually from Australia. So they mention some trees and tree pollens that might not be familiar to us, but the overall scope of it is excellent about how bees collect pollen and then how they convert it into nutrition for themselves, because every plant is different and every pollen is different. And so that's why we almost have this food pyramid for honeybees with different kinds of flowers, because if you stand in front of your hive and you look at the bees coming in, you can see some of the bees might have, a bright orange pollen, and some of them might have something that's actually white or khaki color, and sometimes even ruby red. And all those indicate different flowers that have produced these pollen because they want to reproduce, that the bees have collected. And so they have different nutritional levels and elements in them. This is what keeps honeybees healthy, this relationship between honeybees and flowers. And this is kind of interesting too. And if we have anybody out there that's less than 12 years old, have them cover their ears for just a second, because what bees are looking for is food, pollen and nectar. And what flowers are looking for is sex. Just think of the first plants that were wind pollinated exclusively, kind of like pine trees now, evergreen trees that produce lots and lots of pollen and then express it in the air, throw it in the air. Hopefully this male element of the pollen, which contains sperm, will float around and get to the right place on the other flowers of other evergreens, so that they can produce a seed and a pine cone and what have you. Well, sometime in the past, those plants decided, well, this is pretty inefficient. I'm producing tons of this pollen, throwing it into the air and hoping it gets to the right place. Why don't I make a pact, have a relationship with that insect over there, that I'll provide them some extra pollen and some nectar, this sweet nectar, if they will come over and take my pollen over to that plant across the field, because I really think that plant over there is attractive and I want to have sex with that plant. And so they have had this relationship with a honeybee, two separate species of organisms, an insect and a plant, that have decided to work together for each other's benefit. We're the same species and we can't even get along. But now we have these two separate species that are getting along, and they have been for millions of years, and it has been a positive relationship. I help you, you help me. I get to reproduce by producing seeds, and you get this food that you can eat and feed to your sisters. So this is a tremendous relationship, and something that I don't know that we appreciate as much as we should. And as I said, pollen comes in all sorts of different colors because it has different levels of proteins, fats, amino acids, flavonoids, colorings, and everything else. So each plant produces a different nutritional level. But honeybees do not eat pollen. Pollen is that male element, and it's in a capsule that has a hard coat on it called an exene, because that pollen with the sperm inside of it wants to protect that sperm until it gets to this other flower in the right spot. So it has to be out in the open. It's being exposed to UV light and temperature and humidity changes and what have you. And the sperm is fragile and it wants to be protected. And because it's in this exene, this hard shell, bees can't eat it. Bees don't have chewing, crunching mouth parts. They can't eat pollen. They eat bee bread, which is this pollen that has been collected, and then bees add through excretions some bacteria and yeast and what have you that turn this pollen into bee bread, which is a food product that they can actually access the nutrition from. So remember this, bees don't eat pollen. They can't eat pollen. They eat bee bread. And you can see, if you looked in your colonies, some of these cells that have this pollen stored with, it's kind of like pollen yogurt, if you will, because the bees have added these organisms to it from their midgut and some honey so that these organisms can grow and enter the pollen. But you can see if you've taken out some cells of bee bread that most part, those cells have pollen in it in different layers and different colors. Because remember, we said every plant produces something different. And if let's say Jamie, he might like this, but let's say Jamie, the only thing we feed Jamie is Hershey bars. He might like it for a while, but after a while, Jamie's going to get sick because Hershey bars do not contain complete nutrition to feed Jamie and keep him healthy. And so this is why we have to vary the diet of Jamie to keep him healthy. And the same thing with bees, they do this because they're foraging in about a 2.5 mile radius around the colony and bringing back all sorts of different things from different flowers in that huge area to make their own pollen food pyramid. So this is a pollen grain. Inside that pollen grain is that sperm I was talking about. And so the pollen grain, how does the sperm get out? Well, I told you it's in this capsule with this hard protective coating called an exene, but there's a little gap in it called a micropore or a micropyle, so that when that pollen grain lands on the stigma of the flower, this special part of the flower, that pollen can stay there and the sperm can make its way through that micropore through a pollen tube and go down to the embryo, the seed, and fertilize that embryo and make a seed. And so what happens in bee bread is those organisms that are in the bee bread, in that pollen that's going to be bee bread, those organisms go up through that micropore and start digesting and eating that sperm up there and break open that exene. It's kind of like fermentation. For any of you people out there that are making, beer in your basement or meat or what have you, that pressure forces out that pre-digested sperm that's up in that pollen, and then bees have an opportunity to access that and get the full nutrition out of it without trying to have to break that exene shell. And I want you all to memorize these. These are the bacteria that have been found in bee bread. And whoever memorizes that by the end of my talk will give you a year's copy to bee culture, okay? This is the honeybee's gut. And the reason I'm bringing this up is because I want to show you a couple things. From the top is the crop where nectar and what have you come in, and it makes its way down through the proboscis and vectricious and what have you, and it's digesting in there. Bees have digestive juices just like we do to digest not only the nectar, but this bee bread that we've just talked about. But there's a really cool thing, I think, in the bee's gut called the peritrophic membrane. Think of the bee's intestine as this big container, if you will, and it has a lining in the container, and that lining is called a peritrophic membrane. So what happens is when the bees are eating bee bread, what have you, and it goes into the gut, and these digestive juices come out, this peritrophic membrane detaches from the wall of the gut and kind of and comes down and encloses the bee bread in that area. It's kind of like a mesh bag, if you will, because the digestive juices can still go through it and digest the stuff, and then the digestive material can go back through this mesh thing. But the mesh is so small, and it's designed to be part of the immune system, because those nutrients can go back out of the peritrophic membrane, but some viruses are too big, some bad chemicals are too big, the molecules are too big in order to make it back out. So the bad stuff is contained within the peritrophic membrane. And you've seen this, I'm sure, especially in the spring, maybe. Everybody here has had their car pooped on by honeybees, right? Now, and this is maybe I need to be busier, but I look at bee poop on my car. And have you ever noticed the bee poop on your car? Sometimes it's a splat, or sometimes it's kind of like a thread. The ones that are a thread are the peritrophic membrane that's intact. It's still in one piece. The splat means the peritrophic membrane has been damaged, and it's been damaged probably by nosema, that the spore shoots basically a spear out from it that's a tube that pierces a cell in the gut wall. The nosema sends its genetic material into that cell to make more nosema. And so it breaks a hole in that next peritrophic membrane. And so these peritrophic membranes, instead of being able to contain the residue of bee bread or viruses or chemicals, is full of big holes that viruses and these chemicals can get out of and cause honeybees even more problems. So the digestive system is part of the immune system. And so this is why we need to monitor our bees and what we see more often than we really do. The storage reservoir for honeybees is in fat bodies, and it stores a product called VITELLOGENIN that many of you may have heard recently. That is basically a fatty sugar that the excess food supply is stored in this. And this is what you probably have heard over the last year or so from Dr. Sammy Ramsey, that Varroa mites seek out these fat bodies. VITELLOGENIN is in order to feed on those, because it makes sense. That's where concentrated food resources are for the Varroa mites, so that they can reproduce. And this VITELLOGENIN is super important, of course, as bees get into, August, September, and those bees change from summer bees to long-lived winter bees, and they have more of this VITELLOGENIN stored in order to make it through a long, cold, hard Ohio winter. And so keeping your bees free of Varroa mites increases their VITELLOGENIN levels, which increases their ability to produce hormones and nutrition and calories to make it through these parts of our season where there are no flowers blooming. Worker bees, especially young nurse bees, have glands in their head called hypopharyngeal glands. These glands produce the royal jelly and the liquid food that the nurse bees feed to their baby sisters. The egg, when it hatches after 3 days, is a young larva, and then the next 3 days, all the larvae are fed royal jelly. After that 3 days, the ones that are going to be worker bees are fed a mixture of a liquid food from the hypopharyngeal glands and bee bread in there, because as you know, if they were just fed royal jelly the whole time, they would turn into a queen. But having big, healthy hypopharyngeal glands in their head of these nurse worker bees is super important for the health of all the worker bees. So if the nurse bees have been fed properly, their hypopharyngeal glands will be smaller, and they will not be able to feed as robustly their developing sisters. So this is where it's super important for these nurse bees to be healthy and well and be well fed themselves, so that they can pass on that well-fed ability to their sisters, and then their sisters will be bigger and more robust and be able to go out and forage more actively than they can if they were otherwise. Just a picture of where the hypopharyngeal glands are in the head and other glands, but the bees have a lot of glands in them. The hypopharyngeal gland for the health of the honeybee colony, because all the workers in there have been fed from products from it, is super important. You see how those larvae are floating in larval food? That's important. If they're not floating in larval food, they may not be getting the nutrition that they need to grow up to be robust workers, but not only robust workers, but robust workers that have a long lifespan and not a shorter lifespan, because the shorter the lifespan is of the worker bee, that means somebody else has to fill in for them. And that's where honeybee colonies might not build up as well or store as much honey in order for you to extract or for them to make it through winter. This is what honeybees love. They love flower pollen. That's what they do. They've been developed, they've evolved for millions of years for this relationship that we talked about between them and flowers. Flowers give them a little pollen, some nectar, and the bees help the plants reproduce. You can see how many acres are involved in these honeybees foraging in that 2.5 mile radius. They can go out further, of course, but just think in your own mind, and I can't do this because I don't know, I'd have to get on Google Earth or something, but 2.5 mile radius around your colony is almost 9,000 acres. What's in that 9,000 acres around your beehive? Is it suburban lawns? Is it cornfields and bean fields? Is it woods? Is it forest? Is it interstate? What is it? Because that is what's going to feed your bees, ultimately. And then bees, like I said, if things are tough, they'll go out further, which is 50,000 acres, which gives them a lot more opportunities. For fun, I just picked out a university, and the University of Virginia has 1,682 acres just in the University of Virginia as kind of a direction. That's a lot of acres. This is what honeybees eat. That's pollen grains on this honeybee. She hasn't groomed herself and stored those in their barbicular and a pollen nugget on their back legs yet. But that's what honeybees turn into bee bread and eat. What do honeybees eat? They don't eat pollen, they eat, oh, okay, I can hear you all saying bee bread. Thank you. Honeybees do not eat this stuff. Honeybees do not eat soybean flour. They don't eat dried egg yolks or dried egg whites. They don't eat rice flour. They don't eat sorghum flour. They don't eat any of this stuff that sometimes our supplemental diets are made out of. Honeybees do not go out and look in the chicken yard for a broken egg. They don't go into the soybean field hoping to find some soybean flour on the ground or sorghum or anything else. They don't eat these products. They can't eat these products many times because they can't digest them. 40% of the sugars in soybeans are toxic to honeybees. And just as an example here, and you've seen this yourself, you put one of these patties, on your colony in some way, form, or fashion, and the bees line up all around it, and you think, oh man, this is great. Look at those bees eating this stuff. Well, you know what? The reason they're lined up around there like that is because these patties are so much sugar in them that the bees are licking it off. The bees are licking off, it's kind of like Tootsie Pop. The bees are licking off the sugar and ignoring, for the most part, those particles of soybean flour or whatever else is in there. Most of the particles, just think of how small a pollen grain is. Most of the particles on flour are too big. They're the wrong dimensions. They don't have the right smell. They don't have any taste. This isn't pollen. This is trash. And where we feed bees on the top of their colony, that's not where honeybees find food, is it? Honeybees find food stored in the cells, in the frame, in the comb. What they find on the top bars or anything else where we put these patties, that's where we find trash. And the bees line up there to remove the trash, lick off the sugar, and take the rest of the stuff out and dump it outside as trash. Years ago, when I was the chief of the apiary section in Florida, we did some trials on this, and we put debris traps on the colonies. Debris traps is a box, if you will, with kind of a maze through it that we could see what the bees were dragging out of the colony as trash, because the bees would eventually drop it in the debris trap before they could get outside and drop it. And we found that on the majority of colonies that 70 to 80% of everything that was in a pollen patty was being dragged out as trash. Sucrose is good. Some sugars reduce the lifespan in honeybees as low as 2% melizotose and what have you. Sucrose is probably what you want to be feeding your bees if you're feeding them just a liquid calorie diet. Certainly, sucrose doesn't have proteins, vitamins, and minerals in it, but it does have those calories, that ability to give the bees some energy, whether it's to produce wax to build comb or to store for an upcoming winter. This is a picture of my bees at home. Who here believes that? Raise your hand, please. Thank you. No, that's not. That's a picture from Canada in bees in canola, which bees do very well on. All right, now this is where I'm gonna upset some other people. There are approximately 50 million acres of suburban lawns in the United States. 50 million acres, taking 18 million pounds of chemicals and 10,000 gallons water each above and beyond rainfall to keep them looking like the 18th hole at Augusta. Grass is a resource black hole. Grass doesn't produce flowers. Grass doesn't do anything other than take up space and use up resources, like I said. Just think if we could take 50 million acres and convert 1%, 2%, 3%, 5% of 50 million acres into some kind of pollinator-friendly area. Plant some flowers. You know, those places next to your house, you can still keep your lawn and keep all your neighbors happy and all that other kind of junk, but you can help not only honeybees, but other pollinators and butterflies and bumblebees and what have you. You know, have some access to reasonable food. Or that space up and down either side of your driveway, or that spot in your backyard that you know, nobody sees or what have you. And so what I want to leave with you right now is that honeybees don't eat pollen. They need flowers to produce the pollen in many different varieties of flowers in order to bring back to their colony and then convert to bee bread. That is a food that is more nutritionally accessible to the honeybees, so that their hypopharyngeal glands will be big and produce royal jelly and liquid food to feed their baby sisters, so they're big and robust and healthy, and that they'll forage in about 9,000 acres around their colony, which allows them to access a lot of flowers, but also get into a lot of stuff that might not be good for bees. It might be toxic or what have you. And then if you want to feed your bees supplemental diets, remember that only about 30% of those patties is actually usable by the honeybees, which 30% is better than nothing. But if you're eating your own diet, say you're going to sit down for supper tonight, you know, break you know, your plate into thirds tonight and scrape off 2/3 of it and leave a third there, and that will be your meal. That will be your diet tonight. So pollen subs are not perfect, and you know, it's all marketing at the end of the day. That's why they put so much sugar in those patties, so that the bees look like they're actually doing something when they're dragging it out as trash. And then sucrose syrup is probably better than high fructose corn syrup or anything else to feed your bees as a liquid diet. And the thing also is that we're beekeepers, and that we have a lot of smart older beekeepers out there to use as mentors. You have wonderful organizations that are available that you belong to. That is why you're on this Zoom right now. And so there's a lot of information out there that's correct. I would say that the worst thing that's happened to beekeeping, in the last 20 years is the internet. You can get on the internet and find all the wrong information that you want all day long. The information that you want should come from the USDA or universities or the Honeybee Health Coalition or Project Apis mellifera, not that second-year beekeeper who's now the world's expert in honeybees and has his own website and blog and telling you that green jello fed to your bees will get rid of varroa and everything will be fine. So I hope that I have confused you, made you a little bit angry, and I'm here for you at any time. So thank you very much.
I do have a question from you from Roger. Do different clovers have different protein profiles?
Right. Yes, that's true. Yes. And so white Dutch clover is a little bit different than red clover if we're going to use two different examples. And the thing you have to think about is clover is you want that flower to be small, like in a Dutch clover, because that flower, as you looked at it, is made up of, golly, many, many, many other individual flowers, and it's called a floret. And you can see how long that floret is. A honeybee's tongue is only so long, and for the bees to access the nectar in there, they have to be able to reach it, and they can get the pollen because the anthers are towards the center. But for instance, on red clover, bigger flower, longer florets, and the bees, unless there's a drought and the nectar builds up in that floret, the bee's tongue isn't long enough to get to the bottom of that flower. And so that's not a good resource. So, white Dutch and many, many other kind of smaller head clovers are the best.
Thank you very much. Does anybody have any other questions? Please type it into the Zoom group chat for Mr. Hayes. From Deb, there's a question. Are there summer flowers that you recommend for protein content? Good question.
Yeah, no, that's true, but it takes about, depending on the flower, from 1 to 2 acres of a monocrop flower in order to provide the appropriate protein, vitamins, and minerals and nectar to one colony of honeybees. And so I showed you that picture in Canada of those colonies in the canola field. Canola is one of those unusual crops, mustard, that the flower has adequate protein, vitamins, and minerals and an excess of nectar that can actually support colonies of honeybees. And so this is why, at least in our area, we don't have that kind of resource. And this is why the bees have to travel so far. So you can plant, and I would go back to the previous question. I think the best crop to plant probably is, clovers, of course. You have, echinacea, and you have some other flowers that you can plant. But generally in our yards, our suburban yards or what have you, we can't plant enough of anything in order to make a huge difference. We can make a minute difference, but it's really up to the environment around us and our neighbors and everything else, as all these things kind of add up into one giant resource that is most important. So yes, Clovers because they produce appropriate pollen and nectar, but also because they're a legume. Roots go down probably 10, 15 feet. They produce their own nitrogen, so you don't have to fertilize them. They get water, so you don't have to water them. The roots break up the soil. So they're a wonderful plant. And I hate to say this, I hope nobody's working for, Scott's or any of these other lawn care places. They always show pictures of clover being a weed and dandelions being a weed. And here, get the stuff that kills these weeds. Well, you know what? It's all marketing at the end of the day. Because they want you to have a green grassy lawn, because that green grassy lawn takes 18 million pounds of chemicals and fertilizer and in water in order to keep it looking. And so they're trying to sell stuff. If you want to have a lawn, I would say put a lawn near of clover. You probably don't have to mow it too often, but you're probably surprised the heck out of your neighbors too.
All right. We have a question from Roger. It's a little bit off topic, but in your research, will RNAi ever kill Varroa?
A great question. So I worked on RNAi for about 9 years. And real quick, everybody knows what DNA is. The DNA is the code for you in the nucleus of the cells of your body. It's the recipe for you. But how does that recipe, how does that individual instruction get to a cell site to tell that cell to turn on a protein or turn on an enzyme or turn it off or what have you? Well, what happens is the DNA makes a copy of that instruction, and a helper of our DNA, RNA, takes it to that cell site and delivers that message in order for that cell to start doing something. But you can't have something running all the time, so you sometimes have to turn that off, and that's where RNAi, I stands for interference. The DNA also says, okay, we got enough of this. Let's turn that off. So they make RNAi to turn it off. And so what I was working on with a bunch of researchers was, how could you deliver RNAi to Varroa to turn something off in Varroa to kill, hurt, or damage it? And you can do all sorts of cool stuff in the lab. We couldn't make it work very well in the field at all because we couldn't deliver it efficiently. Think of RNAi or anything else, or DNA or anything. It's basically, protein, amino acids. So to get it into Varroa, you would have to feed it to honeybees who digest most of it. You'd have to give them a huge amount, and maybe a little bit of that RNAi would get into the honeybee. But then we said that Varroa feed almost exclusively on VITELLOGENIN. How do you get then enough of the RNAi from the hemolymp, the blood of the bee, into Vitell? And anyway, you couldn't do it efficiently enough for cheap enough because it costs money to do that. And then you had to produce so much of it in order to get it delivered to the right spot that it just wasn't practical.
Since there's no other questions, I want to thank you, Mr. Jerry Hayes, for taking the time this evening. And
thank you so much again.
You're very welcome. And for everybody here, I have a poll that we're going to send out. This is asking questions. How did you enjoy Jerry's presentation, the length of the presentation, and did you learn anything? So if you'll take a few seconds here and go ahead and fill that poll out. I'm going to go ahead and share my screen. And what we're going to do, we're going to draw a prize. And we have 23 people on the call. So we're going to add 23 to this. And we'll go ahead and generate number 22. So Matt Reisner, you have a bee brush coming your way. If everybody wants to get up and stretch their legs for a few seconds, I'm going to go ahead and get Barb keyed up. And Barb's on. And I have an advertisement running with all the vendors. Dwayne Hathaway has 10% off Hive Body Supers covers. That special goes on till July 4th. April's Greenhouse. And 10 seconds later, Carlin Honey has a promo code for 15% off on his Etsy. So Carlin Honey's got all kinds of stuff. Got a good special going on for Father's Day also. Meyer's Honey with Roger and Judy Meyer for honey and beeswax products. And Mr. Brad Hibbard for the thermal industry in the Honey Blossom Orchard. The drawing, we started off with the bee brush. The next is going to be the Pro DFM, the microbe. Then we'll have the top hive feeder, the hive tool, and then the last drawing will be the $50 gift certificate. And here's the lineup for the rest of the evening. And for you to be on the call, you must be on or present and online to win the prize. And I want to thank everybody for doing the poll, and those will be shared here in a little bit. Right now, over 85% said they enjoyed Jerry's presentation. The length of the presentation, 94% said it was perfect. And did you learn everything? That's up to 98%. So thank you very much for your input on that. Okay, I'm going to leave the poll up for Mr. Jerry Hayes for about another minute, and then I'm going to close that out. Then we're going to start switching over for Barb. All right, we're going to go ahead and get started back up here. If Mr. Dwight Wilson would like to introduce Northwest Ohio Beekeepers.
Okay, am I on, Jamie?
Yes, sir.
Okay, well, I certainly want to apologize for not being savvy enough with the computer to be on the first time. Jamie had the president's talk, but I finally figured it out. And but yes, I do want to say something about Northwest Ohio Beekeepers. We do meet in Pandora High School when the school is in session. I'm not sure how this fall is going to be with the schools. I'm sure the schools are figuring out what they're doing too, but we'll keep everybody in the loop. But we do meet the fourth Tuesday of each month with schools in session at 7:30, and at the west end of the high school. It's a nice meeting room, and it seems it's well represented with the beekeepers, and basically it deals with education and along with getting people on board, what's happening. I want to thank Jamie for putting this all together. It's quite a feat to bring all these people, and thank the folks that are out there listening too. That makes it all worthwhile. I certainly enjoyed Jerry's talk, and now I know that the bee droppings on my truck are typically mostly healthy, thank goodness, and that's a good deal. But other than looking forward to the rest of the presentation. So if anybody would be interested in the Northwest Ohio Beekeepers, they can get a hold of me or Jamie, and we certainly will fill them in on the club and what's going on. But with that, I'll turn it back to you, Jamie.
All right. I am pleased and honored to introduce Barb Bloetscher. She is the state apiarist and a good friend. She has a wonderful job that I would aspire to have one day, maybe. She's going to go over Mite monitoring apiary inspection program. So I'm going to let you take it away, Barb.
All right. Well, thank you. And thanks, Jamie, for putting this together, because I'm sure I know you have a million things to do, and this is very time consuming. So thank you for that. And thanks for everybody for sitting down and watching presentation on a beautiful Saturday afternoon. So Jamie asked me to talk a little bit about the apiary program and mite monitoring. So with that, I'm going to, so I'm the, as Jamie said, I'm the state entomologist and state apiarist for Ohio Department of Agriculture. And I've had this job 11 years, come from Ohio State Extension. So very different kind of job, but it is a good job. I get to meet a lot of wonderful beekeepers, and that's really the best part of the job. So the bee inspection itself, or actually the apiary program, started back in 1904. They had the idea that they needed to inspect colonies to keep the bees healthy. Then around 1934, they finally hired the first state apiarist. If you have trouble sleeping at night, I have links to the laws and rules, which have been updated a few times through the years. And I want to go back in history because I think this kind of explains the importance of the apiary program. So back in from 1923 to 1930, they had an apiary program, but they didn't have any inspection program. So they had voluntary inspectors going out and checking colonies. And the report showed that over 2/3 of the colonies in Ohio were either dead or dying from American foulbrood. So they realized that the voluntary inspection program just wasn't working. And that's when they initiated the inspection program in 1930. And you can see from these old records, cat fur in my face, that back in 1930, we had like a 14% of the colonies in Ohio had American foulbrood, which is seriously, it's a very serious contagious disease. And it was really affecting our honey production and our pollination. Once they initiated the inspection program, within 10 years, by 1940, it was down to 3%. And since then, it has continued to go down thanks to the work of our county inspectors. At this point, we're down to like a 1/10% of the colonies in Ohio have been found to be positive for American foulbrood. We are finding some cases every year, so we try to stay ahead of that. If we do find American foulbrood, we check the colonies in the vicinity to make sure that they're still healthy. This is old data that one of our inspectors, Ed Newman, compiled for me, just showing the shift in the number of colonies through the years. So going back to 1960, we had a lot of colonies. The number of apiaries is pretty much consistent, but the number of colonies has really gone down because people have moved into the city. And I think 70% of all the apiaries in Ohio have 5 hives or left, and they're 5 hives or less, and they're within the city limits. So that's changed our inspection program quite a bit because now our inspectors are doing a lot of urban inspecting, which makes it kind of interesting. You meet more people that way. This kind of shows how the dynamics of where the apiaries are. So the larger the circles, the more colonies there are. You can see up in Northwest Ohio, that's where most of our commercial beekeepers are. And you can see that there's fewer apiaries up there, but many more colonies because they're going to have 40 colonies per yard. Not a lot of beekeepers out there, but they have lots of apiaries and lots of colonies per apiary. And you can kind of see at the urban areas coming up along 71, that's where most of our colonies are. Most of our apiaries are, but not as many colonies. So it's just kind of interesting to see how the apiary program has shifted through the years. Briefly, I wanted to explain what my job is as the state apiarist. First of all, I try to make sure that apiaries are inspected. This is important for many reasons. Not only is it a law, but it helps us keep track of disease. If we do find disease or high levels of, say, a small hive beetle, we can check the apiaries in the area and help the beekeepers keep these pests under control. Where we do have outbreaks of American foulbrood, we check the colonies in the area, and we warn the beekeepers that, hey, we found American foulbrood, and you want to keep an eye on your beehives. People who are not registered, we may not know that they exist, and we can't find the source of the American foulbrood. Also, if your colonies are out in the country, this is a way for the applicators to know where your hives are if they're registered. We try to control and stop the spread of serious diseases, which is really the most important of our jobs. We inspect all the beekeeping businesses that are selling queens and nukes in any colonies that are coming in or out of Ohio, just again to stop the spread of diseases and pests. So our main job is really to ensure healthy beekeeping in a growing beekeeping industry in Ohio. We're not the bee police. We're here to try to get the colonies to be as healthy as possible so we have good pollination. And I say this because I'm happy actually because our total loss over the winter has gone down this last year. We were averaging 50–60% for quite a while. We're down now to about 29%. This map is based on Bee Informed Partnerships survey that they have every year. If you're not aware of the Bee Informed management survey, it's really interesting. It has a lot of useful information for all beekeepers. They ask you about different management techniques and what worked and what didn't. So did you wrap your hives, what your percentage of colony loss was compared to others. They go by state now, so you can look at Ohio and see, okay, these people wrapped their hives, these people used Mite away quick strips, and what was their average loss over the winter. So this has really helped a lot for me to see what techniques work and which ones are less effective. So just to get into this, I can't speak today. I want to talk a little bit about the Apiary program and our rules and the Queen program. Then I'm going to launch into the Mite biology. So we had this, we wrote this law a couple of years ago, and it's mainly to protect beekeepers. And we're still seeing really scary looking nukes. I was inspecting this week, and we saw some people who had, one frame of brood and maybe 2 handfuls of bees, and they wanted to sell those as nukes. And unfortunately, they did not get a queen certificate, and they were already selling them before we even arrived. So it's really an buyer beware. If you buy nukes from somebody and they don't have a queen certificate and you don't look at those nukes, please at least look at them before you take them out of before you give the money away, because you can get a loser colony like that. Those colonies aren't going to do anything. So it's just really important. A nuke by definition should be a queen and all her progeny. So that colony should be this one here on the left, ready to blow, blast off. And you can see there's a frame down here. It's solid capped brood. So this is what you want to see. And that's why we wrote the rule, was to protect beekeepers. Queen production program, this has really taken off. A lot of beekeepers in Ohio are selling nukes and queens. So we, this year, especially because of the COVID, we sent the queen certificates out to everybody who was issued one last year, just to try to speed up the progress, because inspectors have been really limited on being able to go out and do inspections. But I urge you and encourage you to ask for that queen certificate. There are people that who probably have great bees, but they were denied because they had disease, European foulbrood or nosema or whatever. So if they don't have a queen certificate, might be a reason for that. So please remember to ask for that queen certificate. And again, I encourage beekeepers to tell us that they want to sell queens and bees so we can issue you a queen certificate. Whoops. So apiary inspectors in general, they're paid by the county. Every county pays differently. Some pay better than others. Some pay by the hive, some pay, by the hour. They have a lot of paperwork to do, but they're really limited on how many trips they can make to a certain area. So we ask that beekeepers try to work with the inspectors because they can't make multiple trips, across the county in the same area. They have to cover the entire county with the limited funds that they have. We get to a yard, we think they have 2 hives, and we get there and they have 20 hives. So it takes a lot more time. So it's hard for us to schedule a time or even sometimes a day to do inspections with somebody because, every stop we had problems there, or the beekeeper had lots of questions. And we like to help the beekeepers. It's one of the reasons we're there, is to help the beekeepers understand and do better and have a more successful experience keeping bees. So it's hard for us to have any kind of a schedule. They're very important. They're my eyes and ears, and I couldn't do this job without them because I certainly can't cover the entire state by myself. So the county inspectors, they monitor for pests and diseases. They let me know what's going on in their particular county, what problems they may have. They do a lot of teaching while they're out the yards. They do demonstrations. They show beekeepers how to solve whatever problem they may be having. This spring, they did a lot of post-mortem diagnoses to try to determine why somebody's bees died. They observe trends, and they're often the ones that see a new problem in an area. So some counties have inspectors, some don't. I think there's 10 counties with no inspector. If you want an inspector and you don't have one, please contact your county commissioners and encourage them to appoint one. Also let me know, and I can try to make a point to come out and inspect colonies that are in counties with no inspector. But they're very important to the whole program to keep track of pests and diseases. So they're very important, and probably beat down some days when it's hot and sticky and they get stung a lot, but we can't have healthy bees without them. And we're very lucky because some counties or some states have totally eradicated their program. So I wanted to again launch into the mite and their problems and management. This is a picture. A lot of my pictures were sent to me by bee inspectors, so thank you for that. You can see there's a mite on this bee's thorax. This one's got deformed wings. Varroa mites affect bees in a lot of different ways. You can see this is broodcomb over here. All those red dots, those are all varroa mites. So those mites are chewing on those bees, larvae, or pupa. They are not going to be able to develop correctly because of that heavy mite pressure. You can see this is a drone. You can see very clearly see the mites. So we can all see these mites if we look for them. So these are the mouth parts, the chewing mouth parts on a mite. You can see earlier they thought that mites were sucking on the blood of the bees. Actually, what they're doing is they're chewing. They actually chew through the exterior surface of the bee, and they're eating the fat bodies within those bees. So they're doing damage to the exoskeleton of the bee. This is just kind of a cross section. This is a bee segment here. This is her back and her legs down here. These mites, what they found out, Dr. Sammy Ramsey did a lot of research, and they found the mites are actually burrowing underneath the segments of the bees, and they kind of latch on with their mouth parts and their front legs, and they can get a really tight grip in there, and they're not going to let go. So it's, they're doing a lot more damage than we initially thought. And this is kind of a complicated slide, but it does show some of the types of damage that they do. So nutrient storage and mobilization, they're not able to store amino acids, which is important for growth. They have deformed body parts because they can't metamorphosize correctly. water loss because something has chewed through their exoskeleton. They can't maintain their moisture level within the body. They have higher overwinter mortality, or decrease in their lipid and amino acid production, which we have in our own bodies. VITELLOGENIN, that is basically the protein that they need to be able to develop as adults. So these are all different things that mites are directly causing damage to the bees. So just so you can see what the mites look like, obviously these are enlarged. Mite, you don't see those except down in the cells. They never leave the bee cell. And this is kind of a diagram showing the progression. So the queen is laying eggs in here. The worker bee is feeding the larva. You can see there's a mite on that bee. That's a female mite. She crawls down in the cell. And I always get this wrong, so I'm going to show you this slide. She crawls down into the cell before it's capped. She goes down and burrows down in the royal jelly, which is meant for the bee larva. The first thing she does is lay an egg that hatches into an adult male, hatches into a male mite. Then she'll lay more eggs. Every 30 hours she lays another egg, which develop into female mites. So the male and the female mites actually mate down in those cells. The mother mite, actually she chews a hole in the bee pupa, so the young mites can crawl in there and start to feed. The mom starts to defecate on the edges of the cell so the mites can crawl up to get a better grasp and get the bee in different areas. Drones obviously are preferred because they survive they live as a pupa a few more days, so they can get another couple of mites that develop off of that. So the whole life cycle from egg to adult mite is 6 days. So basically every week, you've got another whole generation of mites. So in 3 weeks, your population of mites triples. And that's why it is so important to keep our mite level down. Because even if you treat for your mites and you get 90% success, that extra 10% in 3 weeks, you're going to be right back up where you were before. So I know it's kind of a messy slide, but I wanted to show how important it is to keep our mite levels down. And what the research has shown is that the mite population actually is the highest in the fall. This is important for several reasons. So the bee population starts to decline as the season progresses. We go into a drought, the queen slows down on her egg production. So you have fewer bees per colony. At the same time that the bee population is declining, the population of mites actually increases. And in addition to that, as colonies are failing, the bees are basically robbing, honey and such from other colonies, and the mites from the weak colonies are hitchhiking on the bees and coming back. So you can see, October, November is when our mite population is the highest. When I started keeping bees back in the 80s, we were taught, you treat once in the spring and you treat once in the fall, and that's it. And we can't do that anymore. We have to be monitoring our mite levels really from July on to keep that mite population low. And we have to keep monitoring our mite population through the fall. You think about our last few falls, it's been warm in December. So nothing's flowering in November, December, and the bees are still flying. The only way that they can get food is to rob from each other. So that's one of the reasons why our mite population is growing in the fall, is because they're being transported from one colony to another via robbing. So these are some Beltsville results that came back a couple of years ago, but I found it amazing that these samples were taken in February. So the colony was dead, and they sent in their dead bees that were still on the comb, and the circle's in the wrong place. On the one down here, 120 mites per 100 bees are on dead bees. This one here, again, this is February, 40 mites per 100 bees. So even the mites were still alive. Even after the bees have died, that mite population, one, is extremely high, but also the mites were still in there. One of my inspectors told me that he had some dead outs. He bought packages and put them in there, and he had checked the mites, checked the bees for mites before he put them in there. He treated the packages so they had no mites. He put the packages in there on top of these dead outs, so he had not dumped out the dead bees, and they immediately had high mite population. So the mites were still alive even though the bees were dead, and that still astounds me. But I think that this number is so high because the bees had been robbed so badly in the fall. So the problem, not only do we have, the direct results of the mites feeding on the bees, but we also get problems like PMS, or parasitic mite syndrome. And what this is, so the mites have dirty mouth parts. They're hitchhiking and jumping off on flowers and dead colonies and everything else. They have dirty mouth parts, and as they're feeding on these bee pupa or bee larvae, either one, they open that exoskeleton, and other microbes get in there. There's other fungi and bacteria that are in a colony. They get in there and they infest these bees, young bees or developed bees. And first thing you know, they turn to this goo. So this isn't a virus when it looks like this. I mean, it's not a foreign wing virus or something like that. It's not a bacterial disease when it looks like this. When they turn into this just yellowy goo, that's because of varroa mites. So often we'll go to an apiary and we start seeing this. The beekeeper has treated for mites right before we get there, but we're still seeing this because the mites had already caused the damage to the bee larva. So it takes a while for these bees to come out of this when you've had a high mite population. So if you wait until fall to treat for your mites, bees, developing bees already have these bacteria in them. They're already affected. And at the very time when we need every single bee to survive the winter, these developing bees have been affected by viruses or PMS or whatever. And you kind of look in this lower picture, these pupae have actually been chewed out. Bees were removing them. You can see down here, these have lost their shape. These had all died from PMS. There's another one up here. That bee had undergone her complete metamorphosis. She was ready to be pulled out, and she died with her mouth out, or her tongue sticking out. So I pulled that bee out, and there was actually a mite on her. Her abdomen was completely shriveled. So she didn't have the energy to pull out. So just more symptoms, scattered brood pattern, because as the pupa died, the bees are pulling them out, and just regular pattern. And obviously there was mites or viruses that cause this as well. And as well as the PMS, we have viruses. So this is a comparison of Ohio's mite and virus levels compared to the national levels, and you can say we pretty well match up. So our mite population increases. It's the highest in September and October. It starts to go down. Obviously as the bees start to go down for the winter time, we have I think 23 different viruses that bees get. We don't have them all in Ohio, but we pretty much have this same level of viruses other states do. So the best thing that you can do to keep your mite level down is look for them. Don't just treat without checking your mite level, because you don't know how well you did. You don't know how well that treatment worked. You don't know how many mites you had. So again, you could have a 90% control, but that doesn't mean that 10% is going to build right back up again. So it's important to at least once a month inspect your bees and check for mites. This is looking down into a cell, all this white stuff here. So here's a mite here, and there's a mite hasn't hardened off yet. A couple more mites down here. Scott Schwab, another inspector, took this picture. This is all mite defecation. These are mite. So the female mite, the mother mite, she poops on the side of the cell for the mites, young mites to crawl on. This is a really bad, severe infestation. Here's a drone here with its purple eyes. When you look in your cells and you see all this white dandruff kind of stuff, granular stuff, that's mite poop. You know, it's where the mites have defecated, feeding on the bees. So if all that stuff came out of a developing bee, you can about imagine that that bee is not going to live as long. Their lives can be cut up to 3 or 4 days from that, and they can't function correctly in the hive. Jamie, these are not Jamie's bees. This is a sample that Jamie took. That when we were together, we were sampling somebody's colonies, 300 bees, and we got about 45 mites. So that colony, it was still alive. If you just look at that colony, you thought, oh, the bees look fine. They were suffering. They had severely high level of mites. They had other problems as well. But if you just treat for those, you're going to knock some mites off, but you're not going to kill them all. So I don't know if those colonies survived or not, but they were in really bad shape. So there's basically 4 different ways to check for mites. The easiest way, and some of the inspectors do this just because we're only in a yard once a year, is just pop off some of the drone pupa and look at the bellies. You're going to look at this area. I mean, this is exaggerated. This is terrible infestation. But you look at the percentage of drones that have a mite on them. You can do screen bottom boards. A lot of people do that. All these red circles on here, these are all for mites. So it shows a trend. If you don't know how many frames of drones you have, frames of brood you have, you see that your mite population is going up. It doesn't tell you what your percentage actually is. So they're okay, but they're not great. A lot of people don't insist on sugar rolls because they don't want to kill their bees. Well, a queen lays about 2,000 eggs a day, and you lose about 1,000 bees a day just by old age. So, sampling 300 bees is not a big deal for the colony. If you did it once a month, or you can switch back and forth, use 2 different techniques for sampling, but the sugar roll is not as effective. It doesn't really tell you how well that colony is or what that mite population is. And this is why. Again, these mites have shoved themselves up underneath these segments and they're hanging on with their mouth parts and their tarsal claws. A sugar roll is not going to make those mites drop out. They're going to hang on. So that when they discovered this, this fact about the mites, it blew the effectiveness of sugar rolls. So really the best thing to do is to use an alcohol wash. It does kill the bees, but it kills the mites. And because of that, the mites come loose from the bees, and then you can get an accurate count. So you want to collect at least 300 bees to get a better percentage of the colony, and you simply divide by 3. So you can use jars like this with a screen lid. You can use different techniques. Some people use 2 jars that they attach together. All different things you can do. I like this because it's fast and it's easy, and I can recycle the alcohol. So this jar, this is Easy Check by Vito Pharma. I usually mix, especially now because alcohol is hard to find. I mix windshield fluid with alcohol just so it spreads further, lasts longer. You want to fill it up so it's just up to this first line of circles here. So this is like a drum in here, and it will spin around just like a washing machine. So you collect your bees, and there's multiple ways of doing that. You collect your bees in this part. You have your alcohol in here, and you shake it and shake it and shake it and shake it. Then you pull this drum out like this, and you can lift it up. I'm pointing with my fingers. You can lift it up, and you can see the mites down in the bottom, or you can pour it out through a sieve and collect the mites. And I forgot to ask you, Jamie, if you're going to show that video or not.
I am not.
Okay. It wasn't that great. It was kind of hard to hear.
Yes.
So We tried, but it was just a little too windy out there. There's a pretty good video here. This is Vito Farmer's website that shows you how to do it. There's another one down here with YouTube. So a lot of YouTubes on how to use this thing, and a lot of them they're way too wordy, way too long, or not really effective. So the one down here was the one I felt was the fastest to watch, the least amount of time to watch, and the most effective, most direct, easiest to use as far as, you know, manipulating your hands and everything. So if you get a chance, check that one out. Everybody asked me what to use. They made it easy. Some people don't want to use chemicals. They'd rather use like drone brood removal or something like that. I can't recommend this more. This is free. It's updated every year. It's written by bee researchers like Dr. Dewey Karen. They go through cultural ways of controlling mites, like doing splits, using drone trapping. They talk about chemical and physical ways of controlling mites, and they do it through the season. So it's easy. They made all the guessing out of it. So you go in there, and I've got too much stuff on this screen. They go through like population peak as the colony is increasing in the spring. They're talking about life far moderately effective. It's requeening, division, top guard, oxalic acid drip. Least effective, that's this one down here, bottom board. Then they go into population decrease in the fall. They go through all 4 seasons of the bee season. And what is the most effective? In the back, they talk about each control technique, whether it's chemical or not, how to use it, how it works, how effective it is. So they've just made it so easy. So I just recommend this. You just go to their website and you download it. The USDA threshold right now is still 3 mites per 100 bees. If you're up to 5 mites per 100 bees, those colonies are dead. They don't know it yet, but they're dead because of all the secondary problems that have affected those bees. They have an app to make it even easier. You go to honeybeehealthcoalition.org Varroa tool, and they kind of ask you questions. Do you want to use chemicals? Yes or no? What season is it there? Yes or no? And they show you exactly what techniques are the most effective. So very, very easy to use. I want to make sure I stay on time. You can go on their website and they have videos on how to use these different techniques, how to sample, how to use checkmate or apistan or formic acid. So they make it easy. They're all pretty well put together videos. So the key for all of us is to have healthy bees. Healthy bees, you're going to have better pollination. You're going to have better survival over the wintertime. And you have a better time. Beekeeping is supposed to be fun, and if it's frustrating because you're losing bees every year, you're losing money, it takes the fun out of it. So that's really the key. So really, for the healthiest bees, and I'm kind of wrapping up here, you want to maintain low mites all season long. You want to try to keep your pest and disease level low as possible. We do have some diseases in the spring, but you should be able to get that under control. We are seeing more European foulbrood, so we need to keep an eye on that and try to get that control before it spreads to other colonies. People don't seem to realize that you need 1 to 3 acres of food, forage, that Jerry just talked about, per colony. So if your colonies are in town, if you have 5 colonies in your backyard and it's all urbanized, you may not have enough food for those colonies. So you can have 5 colonies and get a decent amount of honey. You can have 2 colonies and get a ton of honey. You're not going to get more honey by having more bees because you're limited by the amount of food that they have available. You may need to feed them if we go into a drought, or especially in the fall, you may need to feed them. You want to start feeding them in August to make sure that they're heavy enough going into the winter. Try to monitor your colonies at least once a month. Please work with your county inspector. They're here to help. And read. There's so many books. There's more books written on beekeeping than any other topic other than the Bible. We have two great journals, American Beekeeping Journal and Bee Culture Magazine. Talk to your experienced beekeepers. Look at their bees. There are some self-proclaimed expert beekeepers who have lousy, crummy, horrible colonies. So you might want to go and look at their bees. Say, hey, can I come and look at your bees? It might be an eye-opening experience for you. And watch out for these YouTubes. There's a lot of scary stuff. Anybody can do a YouTube and post it. It doesn't mean that it's correct for us. It doesn't mean it's correct for our area. So just use it with caution. And then last but not least, I wanted to share our website a little bit because we keep updating it. You can kind of switch through the tabs here. You can look at the registration, what our inspection average was last year, information on selling queens. We have best management practices that manage they mirror what OSBA has. And this is important because you can share these with municipalities, cities or townships who are considering banning their beekeeping in town. If they can at least get the beekeepers to follow these best management practices, maybe they will not ban the bees, because we need every colony of bees. So we don't want cities banning beekeeping. We have a direct link to the tools for Varroa mite, so you can get it there. Links to websites. So we have quite a bit of information on here that might be helpful for you. Also, I'm sure you've all heard about the new Asian giant hornet. If you see something or somebody has reported to you that they saw something that looks like this, please let us know, because we want to keep an eye on this. We want to make sure that we know if it shows up or when it shows up. So we have a reporting tool. You can take a picture and send it to us, and let us know if you have it. Last but not least, if you see piles of dead bees, if you think your colonies have been killed by pesticide, please let us know as soon as possible. Most of these products, the chemicals, don't last long. They break down in the sun and the wind. So if you haven't been into your apiary in 3 or 4 weeks, it's going to be really hard to track down what happened to those bees. So watch your bees. If they're acting kind of funny or sluggish, see if you can find out if some kind of treatment was applied, if corn was planted recently, and report it, because we are trying to keep track of this stuff. If you do report it, an inspector will come out. Or collect some of the bees and test it. I usually recommend that you collect some bees yourself and freeze them, just in case if the inspector can't get out, or if you want to send them in yourself to somebody. It costs about $300 to $500 to test bees. If we do it, then obviously we absorb that cost. But it might be helpful for you to collect some as well. But call us. We're usually out there within a day or so. So let us know as soon as something happens. This is the time of year that we get those calls. This is the phone number to use. We have an 800 number. Call them, not me, because they will turn around and call me. But it slows them down because they need to have that in their system so that they can tell the correct inspector. So call the pesticide division first, and they'll call me. And I usually try to go out with them and collect bees. Okay. Yeah, and if you can, try to find out, who made the application or where the application was made, so we can get out there. I also encourage you, especially if your bees are out in the country, to register for beecheck.org. You can do that through our website. This is free, it's voluntary. Only applicators can see this. They'll go in there, they can see exactly where the apiaries are. You put your contact information so they can contact you if they're going to be making an application, just to let you know ahead of time. But yeah, other people can't see this, only applicators. So it's just another way for you to protect your colonies. And then I ask that you please watch what you're doing with chemicals in these hives. I hear all kinds of horror stories, and I've seen some stuff. We had somebody spray fipronil on their hives to kill the ants, carpenter ants. Fipronil is highly toxic to bees. So he ended up losing all of his colonies because he did that. So please be careful what you're doing. This is my website, my email, my phone number. I also have the link to ODA, the apiary section, Ohio State Beekeepers, and the Ohio State Bee Lab. I'm home these days because of the COVID, so it's probably easier for people to get hold of me. But please call me if you have questions. I'm just starting to get out and do some inspecting now. So I have lots of websites for information for varroa mites and general information. And again, I'm going to leave this with Jamie so you can access these websites. With that, I am done.
And I have one question for you, Barb, from Linda. What do you feed your bees in the fall when we're in a drought?
In a drought, so keepers are all different. I like to give them sugar syrup 2-to-1 because if it's 2-to-1, they're usually going to store it. So that's my my favorite. I like to use in-hive feeders, so I'm not attracting yellow jackets and robbers.
All right. If anybody has any other questions, please put that in the Zoom group chat. Thank you very much for your time, Barb, and we're going to launch a poll for Barb's presentation. If you will please take a minute to fill that out. I am so passionate about it. After we have Barb off of this, I loaded up the Honeybee Health Coalition, how to take samples. So since we're a little bit ahead of time, I want you guys to understand that it's like going to the doctor's office. Why treat your bees if they do not need to be treated?
Yes.
So we're going to try and I want you to do this. If you know me and my club, I make everybody, when we come into the club meetings, when we meet, I make you put your name and your varroa count up on the board. And it's not to shame other people. It's to let everybody know what's going on in the area. When we see that count going up, what we can do, and we talk about it as a group. The biggest problem in our club is we have too many bees now. We have great beekeepers, not saying we're the best, but we're above the bar of the national laws here in Ohio. So please just monitor your bees every month. At the first of the month, be middle of the end, or just do it when you have all your gear. Go out and do an alcohol wash, and then stir on and take this information. Barb's, if I text Barb or call Barb, she's always there. She's got good information. I'm a county inspector, and we use your resources. Please ask us questions. That's what we're here for. Here's a question from Linda. Who do we contact for honeybee inspections?
If you know who your county inspector is, contact that inspector. You're always welcome to call me. Call the office. So the office number, and I'll type this in. It's 614-728-6373. So you can call that number. Missy is usually there answering the phone. She can tell you who your county inspector is. She can email that inspector and have them contact you. If it's in a county that doesn't have an inspector, she will call me and say, hey, you need to go inspect so-and-so's bees. And I'm scheduled through first week of July because I'm just now getting started doing inspections. So I'm trying to put some schedules in to get around Ohio.
Chris has a question. What was the address to register your hives for applicators?
That is beecheck.org. Beecheck.org.
Yeah, and I have mine registered with BCheck. It's very easy to do. They'll confirm it. They send you back. They constant communications. And for the first time that I've used it this year, I can't remember the program what it was called before, but the applicator in my area, he turned around, gave me a call about 2 days before, and then I turned around and went out and done some precautions as far as my hive. So they are using the program. It's there for us, and it's a good communication that there's not a farmer out there that wants to kill our hives. They want to work with us, and this is a way we can work together. So thank you very much for your time, Barb.
Thank you.
This video will show you two sampling methods step by step: the powdered sugar shake method and the alcohol or soap wash method. Both methods remove mites from the bodies of adult bees, so you can determine the number of mites per 100 adult bees. This is the infestation percentage. If you have fewer than 10 colonies, sample all colonies each time you sample. For larger apiaries, sample 8 randomly selected colonies. If you have multiple apiaries, randomly select 3 to 5% of your total colonies to sample. For both sampling methods, you will need a wide-mouthed jar marked with a line indicating 1/2 cup, a solid lid, a lid with number 8 screen mesh, and a white plate or container. Some beekeeping supply companies sell sampling kits with all the equipment you will need. The only safety equipment you'll need is standard beekeeping protective gear. For the powdered sugar shake method, you will also need powdered sugar and a spray mister filled with water. For the alcohol or soap wash method, you will need alcohol, such as ethanol, ethyl alcohol, isopropyl, or rubbing alcohol, or low sudsing liquid soap, such as automotive windshield washer fluid. For both methods, the first step is to collect the bee sample. To sample a hive, open it and select a brood frame. Look for the queen. If she's present, move her to another frame or set the frame with queen aside. You'll need a sample of about 300 bees. This is a 1/2 cup of lightly packed bees. Collect the sample by tilting the frame slightly and moving the jar downward over the adult bees so they fall into the jar. Or shake bees from 2 or 3 brood frames into a larger collecting container. Then scoop a 1/2 cup of bees and dump them into the jar. For the powdered sugar shake method, add approximately 2 tablespoons of powdered sugar to the jar. Then roll and shake it for at least 1 minute to thoroughly coat the bees with sugar. For consistent results, shake each sample for the same length of time. Set the jar down and wait 3 to 5 minutes. This allows the sample to heat up, which, along with the sugar dusting, dislodges mites from the bodies of the bees. Next, shake the jar like a salt shaker onto the white plate or container. You can repeat the process to improve or verify the accuracy of your count. Add another tablespoon of sugar. Shake again for 1 minute or more. Wait another 3 to 5 minutes. And shake the mites onto the same plate or container as the original sample. High humidity may cause sugar clumping, which can cause some mites to stick in sugar coating at the bottom, reducing the accuracy of the count. Spray the collecting tray with water to dissolve the powdered sugar. Following this, you can return the sampled bees to the hive. This method does not harm them. For the alcohol or soap wash method, take the sample jar away from the colony. Using alcohol around a hot smoker can be dangerous. Add enough alcohol or soap to completely cover the bee sample. Close the jar and shake it vigorously for at least 1 minute. Shaking the liquid dislodges the mites from the bodies of the bees. For consistent results, shake each sample for the same length of time. With a 2-jar sampling device, vigorously wash the bees, drain the liquid through the sample, and count mites. If using a single jar, shake the bees, then pour the liquid containing the mites onto a white plate or tray. Or pour liquid and bees through any filter that separates the bees and mites. The white tray allows you to see and count the mites. Repeat the wash to confirm or improve the accuracy of your count. Add more alcohol or soap to the jar and shake again for the same length of time. This method kills the bees sampled. To preserve the bees, use the powdered sugar shake method. With both methods, count the total number of mites removed from the sample. Divide that number by 300 or by the actual number of bees in the sample if you counted them. This yields the mite level as a decimal. Convert the decimal to a percentage. For example, a sample contains 12 mites. Divide by 300 bees to yield 0.04. Convert that to a percentage, which is a mite level of 4%. To improve the accuracy of your sampling, count the actual number of bees in each sample. As you gain experience, your sample sizes will become more consistently close to 300 bees. The mite levels found in sampling may indicate that further controls are not needed at this time, controls may be needed, or controls are needed now. In general, mite levels below 2% indicate that controls are not needed immediately. Levels above 3% indicate that controls should be applied. Specific thresholds vary according to the colony's developmental phase. See the Tools for Varroa Management Guide for more detailed information on how to interpret your sampling results. Other videos in this series demonstrate how and when to use different Varroa controls. As part of an integrated pest management program, both the powdered sugar shake and alcohol or soap wash sampling methods can give you an accurate picture of Varroa mite levels in your colonies. Remember to sample at least 4 times a year, especially from July through October. Sample again within 1 week after completing control treatments, and use the sampling technique consistently. Thanks for watching this video from the Honey Bee Health Coalition. Visit honeybeehealthcoalition.org and download the Tools for Varroa Management Guide, which has more detailed information on all the topics in this video series. All videos are provided free of charge, and we encourage you to share them.
So no one has an excuse that they can't do a Varroa. If you're looking for the Varroa guide, you can go and download that PDF. If you would like that book, I ordered 100 for my own club. Reach out to me and I will send you one free of charge. I want everybody to monitor your mites. What we're going to do now, we're going to pop up. We have 30 people on the call, or 30 people that have been numbered so far. So our next prize is the Pro DFM. It's the micronutrient feed, and we have number 19, Linda D'Angelo. If Linda D'Angelo is still on the call. Miss Linda, are you there? Yes, she is. And Barb has an excellent comment. For those of you that have either foundation or are making bees that build their own comb, feed, feed, feed until all the comb has been filled out. And since a nectar flow is going and you're trying to build these bees up, remember this is the perfect time to get nukes ready to overwinter, get things built up, do an alcohol wash. And I'm going to agree to disagree with the Honeybee Health Coalition. I would much rather inspect my mites or my mite load once a month. For me, I do it at the beginning of every month. I pay my bills. I do everything. It's kind of bad for me day. So I might as well go out and check my mites and be happy if that I have them under control.
Jamie, I'm going to take the liberty to agree with you on that. I think once a month is a great schedule for checking mites, especially because, as they said in that video, you want to be consistent in your sampling. So that practice, if you're, I mean, a lot of times I'll do a mite wash even in April just to get back in the habit of doing it after a couple of months of not doing it. And it helps with consistency, which helps with accuracy.
Absolutely. The reason I did this lineup is because I hear the biggest excuse to not checking for Varroa. So we went from nutrition with Mr. Jerry Hayes, talking about feeding and looking at your brood and knowing exactly what's going on with the bees. We went into Barb, who's checking on Varroa, and the whole inspection program and knowing who your support system is. And that's the biggest part. And the biggest concern I have with people that say that I don't want to do an alcohol wash because I can't find the queen. Guess what? I have Dave here who has an excellent class and trying to find that queen, double and triple checking, because the girl just does not fly real well or produce baby bees and lay eggs after she's been alcohol washed. So it's a talent. There's a skill to it. And I think Dave has a really good input on this, on how to do this. So I'm going to turn this over to Dave and Mr. Redbeard Bees. You can go ahead and take it away.
Great. Well, thanks to Jamie, for you and all of the bee clubs that put this together and everybody that came together with that. And I am very glad to be a part of this because exactly, nobody wants to be alcohol washing their queen, but we all want to know what our mite levels are. And being able to find the queen reliably and efficiently is a skill that can be learned, and it will take some practice. But let's go ahead and get into this and get right going with this. So finding the queen. What I'm going to talk about today is we're going to talk about a search strategy because it is a skill. There's several parts to the skill. We're going to set up some protocols and some practices that will help us find the queen more quickly. We're going to isolate the queen. We're going to control the amount of light getting into the frames, and we're going to establish and execute a search pattern of the hive. So we're going to be very systematic about it. We're also, and this will be the part that takes a lot more practice, that search strategy and protocol, it's just a simple set of procedures to follow. But you're going to have to train your brain to see the queen. And this sounds like it might be difficult, but honestly, with a little bit of practice, it becomes easier. I used to, when I was first teaching this class on how to find the queen, I would always say, finding the queen is a lot like riding a bicycle. You don't know how to ride a bicycle until after you've learned how to ride a bicycle. Okay? So it's hard to find a queen until you've already found a queen, which sounds a little bit contradictory. But we'll get into it. And honestly, it is a matter of practice that helps a lot. Okay. So real quick, I just want to go over, I think Jamie hit on the number one reason for wanting to find a queen is because we want to safely be able to monitor our mite loads without hurting the queen. But there's other reasons. If we want to requeen, especially if we've got an underperforming or perhaps a nasty queen, we want to get rid of her. Making splits is another one where you need to know where that queen is. Got to have eyes on her. And if we're combining or equalizing hives, trying to add some bees to a weaker hive to get it beefed up for winter, you don't want to add two queens to one hive. Diagnosing swarms, if you get in there and you don't know if your hive has swarmed or not, well, if you find an unmarked queen, that's probably a diagnosis. And a little bit of a look forward, practicing finding queens with unmarked queens is a huge benefit if you can do it. Okay, and just the general health of the hive as well as practice. Honestly, anyone who's ever spotted a queen as they're going across the frame, whether it's your first queen or your 5,000th queen that you found, the thrill is still there. You get that little burst of adrenaline, and it's always exciting. One of my favorite things to do. So first I want to talk a little bit about the queen in the hive. Let me see if I can get my little annotation spotlight out here. So here's our hive. I personally use all medium boxes. So this is my representation of the hive. Here's our bottom board. Here's our brood, the brown, and the red around that is the pollen. And then of course the honey and the outer cover. Okay. Stop that. So the quantum queen, what do I mean by that? Well, you know, there's some 60,000 bees in a colony and one queen, and she can be anywhere in that colony. Okay. So what I like to say is the queen is in every box and no box until we actually put eyes on her. Until our eyes are on that queen, she can be and is in every box. Okay. So she could be right in there where the brood is. She could actually, I've seen queens go from a bottom box 3 or 4 boxes up in just a matter of minutes. As I'm restacking the hive back together, one time I found the queen right up at the top to greet me after I put the last box on. Okay, so she could be in any of those boxes. Another thing to know about the queen is she will hide from light. If you think about the inside of a hive, light is not a natural part of that environment. Okay. Those young nurse bees and queen bees are not used to seeing daylight, and they will tend to move away from it. Daylight will, I'm going to put this in air quotes, it'll stress the bees a little bit. Okay. And similar to the first thing, even when we separate out the boxes, the queen is going to be able to be on every frame and no frame at the same time until she's observed. Again, just that idea. The queen could be anywhere until we actually see her. Okay. So that's what I mean by that. So how do we combat these things? That light is going to push the queen away from us and that we really can't be sure which box she's in until we find her. Well, here's what we're going to do. Here's the answer. First, we're going to separate the boxes. Once we separate the boxes, the queen cannot move between them. So now she is stuck in one of those boxes, okay? And if we're not actively pulling frames out of a box, we're going to keep those boxes covered. And what that does is it maintains the status quo inside the hive so that the workers, as well as the queen, just carry on doing their job. They're not trying to hide from the light or anything. This is a good idea just in beekeeping in general. The more you keep the boxes covered, the longer you can sort of work inside of a hive, because no one box is exposed to light for very long that way. And then the third thing is we're going to leave a gap between the frames when we search through this. Okay, so I'm going to go through these one by one and explain exactly what I mean by all of these. One more tip I forgot. When we get ready to do our search strategy, we want to make sure, right, the queen, statistically speaking, is going to be in the brood nest, because the queen and the brood are basically the same thing. The queen makes the brood, and so that's where she's most likely going to be. So I always like to say, target your search strategy where the queen most likely is going to be. You know, focus your search where she's going to be. I almost never search this top honey box first. Okay. So I want to make sure that I'm focusing on the brood. It's not a guarantee. The number of times I found a queen on the outside wall of a hive, on the inner cover of a hive, up in the honey super doing who knows what. But that's where she's most likely going to be. Okay, so let's take step number 1. Separate out the boxes. If you've got a small hive, it's just building up, you don't have 5, 6 boxes on it, this is easy. You just break all the boxes apart, drop them down, and then we're going to bring in, our queen is going to be probably in one of these 2, but possibly she could be in that honey super. But let's focus on this brood because that's most likely where she's going to be. And let me see. Okay, I'm having a problem just forwarding the slide. There we go. Okay, sorry about that. So there, I've just covered up this middle box. Let me get my annotation up here. I just put a cover on this box. I happen to leave either extra inner covers, or I've got pieces of plywood that I've cut to the approximate size of an inner cover, and I leave those out in the bee yards. And that way, whenever I'm working with bees, if I've got more than 1 or 2 boxes open, I can cover the ones that I'm not working in. Okay, so what do we have here? We have this box where the queen probably is not covered. We have this box. I'd say there's a 50% chance the queen's in there and it's covered. No light getting in. This box is open. And so we're going to go through this box frame by frame, looking for the queen, and we'll talk about how we do that. And then once we're done, let's see if this does. Oops, I apologize. I'm going to go back. Once we're done with that, then we can start putting this hive back together and move our boxes back together. But let's look. We're looking. This is the looking down into the box. I use 8-frame mediums, so these are my 8 frames. Okay, bring the annotation up again. Okay. All right, so these are my frames that I'm looking at through the top of the box. And what I want to do is I want to leave room in between these frames when I'm searching. And there's a reason for that. I'll bring one frame out and I'm going to look at it, and I'll tell you how we search that a little bit. And then I'm going to leave it outside of the box. I get those frame rests that hang on the side of the box. Absolutely love them. Great piece of equipment that I really rely on more and more every year. I'm going to bring a second frame out, and after searching it, I'm going to leave that. We've got now a 2-frame space. I honestly usually take 3 frames out. Okay, but we'll just show here that I'm going to search that frame and move it to the side, leaving a space. Then I'm going to pull this frame out and search it, move it aside. Then I'm going to search this frame, move it aside, and notice here's this gap. What is the point? As we've said, it's going to prevent the queen from going from one frame that we've already searched to a frame we haven't searched. Okay, queens move much quicker than we might think that they do. And also, a queen can go from a frame that's not been searched and go along the wall over to a frame that has. Not common. I'm not saying it happens all the time. But everything that we do to prevent and make it easier to see that queen is going to help us in our search for the queen. Okay, so let's get back to our frame here. So here's our frame. Right there's our brood on there, and there's our pollen stripe, because these bees are awesome and they make that perfect rainbow pattern that we see in the textbooks. And I like to divide the frame. Again, I use mediums. For deeps, you just extend this pattern into sort of like 2-inch squares. And what I'm going to do, I'll show you quick with my pointer, is I'm going to start always search in the same way. The more you can make a habit of how you search for the queen, the easier it's going to be to focus on finding her rather than where your equipment is or what stage you're at. Okay? So I start usually in the top left, and I just scan to the right, down, scan back to the left, down, and scan. So right, just like that. I've tried to time this to be approximately how long I take to scan the frame. I don't spend minutes looking at all this. We'll talk a little bit more about how to train your eyes, but one of the other oxymorons of finding a queen is the longer you look for her, the more your chances of finding her go down. Okay, so if I spend a lot of time on this frame, I'm decreasing my chances of finding the queen. So again, when you're first starting out, take your time, but work towards the goal of trying to do this sort of as quickly as possible. Okay, so again, the timing is, I mean, maybe 5 seconds, 6 seconds. There's no problem with scanning a frame twice. and just do that really quickly. One of the reasons is you kind of want to blur the workers out. So you don't want to spend time looking at workers. Okay. Early on when I was teaching this, I had all the graphics that we just saw, and people were like, Dave, that's all good and well, but you know, it's July and I have 5 boxes, 2 honey supers on my hive. How do I separate out all those boxes? So I put together this little demonstration, and hopefully it helps. It's very similar. Again, we don't have to spread all 5 of these boxes all over the place and have each one open. So what I like to do is I just reverse stack them. So the top goes on this bottom over here, then the next. Okay. Okay. So now I have a stack of unsearched boxes over here that's covered. So light's not getting into it. The queen, maybe she's in here. Heck, she might be going all over the place in here, but I'm going to search this box. Okay. So this way in my bee yard, I don't have 5 open boxes. I've got 1 open box and a reverse stacked hive. Okay. I don't find the queen in there. So what am I going to do? Oops. There we go. I'm going to cover that up because it's already been searched. No need to stress out the bees anymore. And I'm going to pull this box off of the unsearched stack, and I'm going to put it on its own stack here. I am not going to put it onto this stack, because if the queen's in here and I put the box up here, she could immediately go down here and I'll never find her. Okay. So we have 3 stacks is what we want as we're searching for a queen. Our searched stack, we are now searching stack, and we are still to search stack. That way we keep all the boxes segregated, and it just vastly increases our chance of finding the queen. She can't move between searched and unsearched. Okay. Didn't find her, so we'll put it back and we'll search here. Okay. And we're going to say this queen is one of the well-behaved queens. She's not up in the honey super. We found her, so now we can cage her up. We can put her off in a little nook while we take samples for our mite test. We can split the hive, do whatever we want as a result of having found her. And then we'll just put the rest of the hive back together. Did that in a little bit of a strange order. So even if you have a large number of boxes, this idea of separating them out doesn't mean you have to have your hive spread out all over God's green acres. You can just make sure that you develop a habit and a protocol of having a stack of boxes that is not searched and a stack of boxes that has been searched. And in between, you have the box that you're currently searching. Okay. Hopefully that's fairly well straightforward. Okay. Now, that is I don't want to say the easy part of finding the queen. That's the part of finding the queen that takes the least amount of time to get practiced at. All you have to do is just remember those simple steps, keeping spaces in between the frames, keeping your boxes separated, searched from unsearched, and you just have to build that as a habit. I honestly, at this point, I don't even think about doing that anymore. It's just such an automatic muscle memory that even when I'm not looking for the queen, about 90% of the time, that's how I go through my boxes. Okay. Habituating, getting good habits is an excellent part of beekeeping. Now the trick is, how do we, even though we've got this great protocol for searching, how do we find this one bee in 60,000? Well, we're going to train our eyes is what we're going to do. And this takes a little bit of time. It takes some practice, and that's fine. And I'm going to tell you something that's controversial and possibly confusing, but I'm hoping it'll make sense. Don't look for the queen. If you look for the queen, that means you're going to be trying to look at every single bee and determine, is that the queen or not the queen? Just say that 60,000 times. Is that the queen or not the queen? Is that the queen or not the queen? It will take you a long time to find the queen if you're looking for the queen. What I want you to do is approach this as looking for not a worker. Here's the part where if it's not a Zoom call or a Zoom class, I'll open it up to the audience and ask, how many of you, as you've been looking through your hive, you get that little spark of excitement like, oh my gosh, I just saw a queen, almost to immediately realize that what you're looking at is a drone, okay? And then you get sad and disappointed. Oh, I thought I was finding a queen. Here's the good news. If you've ever had that experience, and I'm pretty sure I would guess 90% of you have, if not more, you're halfway to being able to find a queen by looking for not a worker. Because when your eyes perk up on that drone, what they're doing is saying, that doesn't look like everything else. Hey, what's that? That's unique. That's different. It's not a worker. And we want to take advantage of that. We really want to take advantage of that coming in to our search. Okay. So another way to look for not a worker is to see patterns and not bees. Okay. So workers are pretty uniform. One worker has the same basic shape and behavior as another worker. Okay. And so let's talk about that a little bit more. And of course, I'm going to put that in all the time. Practice, practice, practice. I don't expect any of you to go out this afternoon after the field day is over, open up a hive and find a queen within your first 5 minutes of being in the hive just by following the protocol. It is going to take practice, but I hope some of you will go in and find a queen within the first 5 minutes. I think that would be great. So patterns. Let's talk about patterns. Our brain sees patterns all the time. So we want to take advantage of that when we're talking about finding queens, or should I say, when we're talking about finding not a worker. Okay. So for example, here's the moon and here's a potato chip. And our brains are so good, our brains desperately want to see patterns. We just need this tiniest hint of a pattern. And guess what? We can see a man in the moon, right? And we can see Abe Lincoln in that potato chip. So, and you've seen it all, spend a rainy afternoon when you can't get in the bees, Googling potato chips that look like celebrities, right? And you'll see all these things and you're like, how can that possibly look like a celebrity or a person, something other than a potato chip? And it's because our brains want to see patterns so desperately. So we're going to exploit that. So here's our honeybee casts. Up at the top, our drone, and in the middle, our worker, and then our queen. And if you notice, when you look at this, and I should have overlaid these, but this thorax here on the queen and the thorax on the drone are almost exactly the same size. And that's a big reason when you're going through that frame and all of a sudden you're like, oh, is that the queen? Oh, no, it's a drone. Oh, I'm no good at finding queens. You're recognizing, I believe, that thorax as being the same size as the queens, which means you were wrong when you said you were no good at finding queens. You're much better than you think you are. Okay? But we've got to, let's simplify these. Let's simplify these cast shapes into something like that our eye won't get so carried away with all the details. So this is how I like to think of looking at the casts. Drones, even though the thorax is very much the same size as the queen, there's some differences. First, the drone is very much like a squat rectangle. And the thorax of the drone and the abdomen of the drone are so close together, whereas with the queen, there's this little space between her abdomen, and her abdomen is long and extended and comes to a gentle point at the end. And then there's workers that are right in between. Another important thing to think about, workers, the abdomen is basically the same length as the wings. Whereas the queen her abdomen is almost twice as long as her wings, right? So these are the shapes. Here's my trick. Here's how I like to think of the queen. I like to think of her like, right? Everybody loves the queen. The queen's exciting. And when you see the queen, right? She is like an exclamation point. She's like, look at me, I'm the queen. And her body honestly looks like an exclamation point, right? So that's what I'm kind of looking for. And if I tell my brain I want to find an exclamation point, even if I'm looking at a potato chip bag, I'm going to find one, right? Okay. So let's get into this. Patterns, not bees. Okay, step number 1, make sure you train all of your hives to line up really nice and neat, just like this picture shows, and it makes finding the queen so much easier. Okay, couple hours, do positive reinforcement with treats. No, I'm joking. Obviously, we can't do this. But what I want to do is I want to take these simple shapes to show you what I mean by finding not the queen. So here's where I would ask too, does anybody see the queen in that group of bees? The answer is no, because I'm often a little bit cruel in my teaching and there's no queen in there. But we're going to go through a couple of these. I'm going to go through them slowly at first. Take a second or two to scan to see if you see that exclamation point. If you don't worry, because we're going to go back and go through them a little faster to hopefully illustrate another point. Okay, so take a second or two, see if you can see the queen in there. No, because that's the same one I just showed you. There we go. We're going to have a queen in this one, I think. Yep. So just give you a second or two. Don't worry about it. If you're looking at individual bees, you're not going to find her. You really want to sort of step back, get a little blurry, and see where there's a break in the pattern. The worker bee should be creating a pattern, and you want to break where that break is. Okay? Just like that. And there I switched her position, so she should have really showed up on that last one. Again, if she doesn't, don't worry. We're going to go through this again in a slightly different way. Yep. I saw somebody pointing. They saw it. Good job, Adam. You were pointing at the right place. When the queen when she's actually oriented like an exclamation point, it's much easier to see her. So train your queens to always be head down on the frame. Again, that's a joke. You can't really do that. At least that I'm aware of. All right. So what I'm going to do is I'm going to go, pardon me while I go back a little bit. Okay. Now, on this round, I'm going to go through this a little quicker. And again, if you didn't see the queens, we can come back to this, but I usually spend a lot more time on this during some of the in-person classes. But if you didn't see the queens the first time, don't worry about it. This time, I really don't want you to look at individual bees. I want you to sort of stare at the whole thing. Remember, maybe you remember those magic eye drawings where you had to stare at them for a long time before you saw the 3D image come out. I want you to stare at these. I'm going to click through these a little faster, and at some point, your eye should pick up on some motion, because motion is actually another really important component of finding the queen. Okay? And I'm going to we'll use some videos as well. But I'm going to click through this a little faster, and you might see her if you're just staring at the whole block. Like right there, she popped up. And when she's moving, she's actually sometimes easier to see. Okay. Again, I'm going to reiterate this. We can come back to those if we've got time at the end, and if people would like to go through that a little bit more. But again, just to reiterate, this is the kind of speed that you should be aiming for eventually to scan the frame. Not fast, but definitely not slow. Just a nice, steady left to right, right to left to right, until you cover the whole frame. Now, some people say, you know, is it possible the queen could be right here, and as you scan over, she goes up to this part of the frame, and then you miss her on the frame? 100%, absolutely. I will tell you, just earlier this week, I was out in my mating nooks trying to find a few queens, 5 frames in my mating nooks. And I checked one 3 or 4 times and I could not find the queen. She got away from me every single time. Okay. So again, sometimes you have to scan it twice. Don't be discouraged if you don't find her the first time through the box or the hive. Just keep with it and keep practicing. Okay, so here we have a video. This is one of my favorite videos to share. If you've been to any of my classes, whether they have to do with queens or not, there's a good likelihood that I've shared this video at the end of my class when we've had extra time. Okay, right there's a queen. We can tell by the dot that we put on her. And like I say, it's best to train your eye, and we do have some unmarked queens in videos to look at, but it's best to try to train your eye on an unmarked queen because you're training your eye to see the shape of the queen, not to see the color dot on the queen. So I'm going to play this video. This queen is going to go around and she's looking for a place to lay an egg. The good news, spoiler alert, we're actually going to see her lay an egg, which is always a treat. And after she lays an egg, she gives one of her adult daughters a piggyback ride. It's the most adorable thing, I promise you. But what I want you to pay attention to, and I'll play this video more than once, is notice the queen, how she moves, and it's very differently than how the workers move. So a lot of times the workers will either be still, shaking, or moving much quicker. The queen is going to move in at a very steady pace and kind of like a meandering river. She's not going to be going in straight lines, but she's not going to be jerking all around either. She's just kind of like a nice, steady, meandering river. So go ahead and keep your eyes on the queen. And notice the coloring of her abdomen. Even though this is a striped queen, which not all queens are, it's very different from the type of striping that the workers have. The workers almost have like pinstripes, whereas her abdomen is very gradually striped. Oh, that's a good candidate. And there she goes. And they are just licking and scraping all that pheromone off her. Not enough that she has to lay 1,500 eggs a day, but she has to give piggyback rides as well. There we go. A mom's work is never done. Okay. So hopefully that made sense to people, seeing that when I'm talking about a nice, steady, slow, I shouldn't say slow motion. She's not like, you know, the heroes at the end of a Marvel movie in slow motion, but moving in a nice, steady pace. And she's not zigzagging all around, even though she's not, it doesn't look like she's doing something purposeful. You know, she's not executing a quadrant search pattern. But let's see if I can play that again. Oops. Nope, let's go back. One. All right, we're going to play this one more time for everyone. But notice how the worker bees also kind of, the pattern of the worker bees breaks apart because she kind of pushes her way through, right? So up over here, they're all packed in together, but here they kind of break apart as she pushes through. So that's the other thing I mean when we're looking for not a worker. We're looking for that break in the pattern of workers. And I found a lot of times there's that little extra space around her when she's walking. Yeah. Okay. Okay. Let's go on to one more video. Now, I've already spotted the queen here, but I have probably, you know, I've seen this video however many times, so I kind of know right where she is. What I want to do is I'm going to play this without pointing out the queen. She is unmarked. And then I'm going to go ahead and restart the video and we'll play it. And then that way you can watch her. So use this as a chance to try to find the queen. Oops. And again, I went, sorry, no, back. Let's just do it this way. Okay, there we go. Sorry about that. Okay. Again, we're looking for a break in the pattern. If you're not seeing her, don't worry. This is not, this isn't something that just happens automatically. If you're in the habit of looking at individual bees for the queen, that's a hard habit to break. And actually, at the moment, was completely covered by other workers for a bit. Okay, so what I'm going to do is back up, if it will let me. Nope, it sure won't. Okay, I'm going to have to close this. There we go. Now I can get back to here. Okay, so now I'm going to point out the queen. The queen, I've got the red dot right on her thorax. Okay, so there's her thorax, there's the queen. And again, notice, we'll see at one point she becomes hidden underneath workers, right? But a lot of times there's just this little buffer around her that if you look up here, none of the workers get that buffer around them, right? So there's something about a queen pushing her way through, or the workers make the way for her. So I'm going to start this. This time I'm going to try to follow her with this red. I'll keep the red right by her. And then we'll play it another time where we just let your eyes follow her. There we go. And again, her motion, if you look at the workers around her, it looks like they've all had a little bit of caffeine. They're just a little bit shaky. And right there, you're never going to see her if that was what you looked at, because she was underneath, all of her daughters. And there she's a little bit there, boom, covered up again. Okay. So this is why I don't want you to be discouraged, because there are times, even if you're doing everything 100% absolutely correctly, you may just have put your eyes on her at that point when she's under 2 or 3 of her daughters. Okay. So let's go ahead and I'm going to play this again. This time I'm not going to keep the red there. Remember, she starts out right about in the middle. Right there she is. And then just follow her as best you can. And again, she's kind of pushing her way through and breaking up those workers a little bit. Even though she goes under them, they tend to move off and away from her more quickly than if they're gathering up on other bees, okay? So it's a combination of things. We're trying to sort of blur the workers out of our vision to make the workers all seem like one background pattern. And so we're looking for the thing that's not a worker. I used to have photos that I'd use like flashcards. And in one of the photos, I took a photo of a frame that was probably 80% drones with one queen on it. And I did that again, just to be mean to my students. Okay. So here, in fact, this is a video where there's actually some drones in here, right? Drones. And I haven't spotted the queen yet myself, but I'm going to go ahead and hit play. And when I find her, I'll put the red dot on her. And hopefully some of you will find her before I do. Okay. Right there she is. Yep. But again, notice that thorax too. It just really, really shows up. Unlike the drone's thoraxes, it's not hairy. She's right in there. Now, something else you might notice about that queen, that is not the biggest, plumpest, fattest looking queen that anyone's ever seen. That may be a virgin queen. I can't remember if that's a virgin queen or a newly mated queen. Which brings me to a good point. You want to start, you're practicing. This is a good time of year to start practicing. You want to start looking for nice, plump, healthy, well-mated queens because they're bigger and easier to find. If you've got a virgin queen in there, they look very different from mated queens. They look very similar to workers almost. They're very tiny, and one of the biggest differences is that their abdomen comes to much more of a point. So if you've got a hive that you think has a virgin in it, I wouldn't start out searching for queens with virgins. Don't do that to yourself. Start with what's normal and what happens 90% of the time. A nice, big, plump, healthy queen. Also coming out of winter, even a 2-year-old, 3-year-old well-mated queen, she's going to look very different than she does in June and July. Again, she changes as her job changes. In the winter, her job is to stop laying eggs, and in the summer, it's to be laying eggs. Okay. I'm just checking here to make sure that there weren't questions typed in. Okay. All right. That kind of brings us, Jamie, I'm not ending early. That brings us to the official end of the presentation. That covers all of our top points, right? Which we will go through again, but I have a pro tip that I want to share with everyone first. So what if I need to find a queen because I'm doing a split or I'm requeening, okay? And this is sort of a preview of some of my requeening classes. So let's say I'm just gonna requeen this hive. And I happen to know I've ordered some queens, and they're going to show up next week on Tuesday. So what I want to do is I want to make sure that I can find the queen, and I may even cage her because I think the easiest way to requeen is to take one cage out and put the other in. So I want to start looking for the queen at least 4 or 5 days before I'm going to replace her. And I'm going to explain why with this pro tip. I followed all of my protocols. I separated out the boxes. I kept the boxes covered. I kept room in between the frames as I was searching, and I didn't find the queen. What do I do now? Dave, your protocols just didn't work. They failed us. Don't worry. We have a backup plan. I think we would all agree that beekeeping is all about having good backup plans. Okay. So here's my pro tip. Couldn't find the queen? Even if it's 5 boxes, whatever, I'm going to use queen excluders. I actually use queen excluders for manipulating queens more than I do for keeping them out of my honey supers. So I've got 2 queen excluders here. I'm going to open up my hive. This shouldn't take more than maybe a minute or 2 at most. You're not even really opening the hive. I'm going to put my queen excluders in, close up the hive. I don't even take a frame out. And I'm going to come back 4 days later. And what is the advantage to this? I now will only have to search in 1 box to find the queen. I have reduced the amount of area I have to search by 2/3 or more if I've got more boxes. What I do is I now go in and find out which box has eggs. The box with eggs is the box with queens. And then I can spend all day searching for that queen there. Or if I'm making a split, I just take that box aside. Split done. Don't even have to put my eyes on the queen as long as I've got plenty of brood and food for the remaining hive. Okay? Let me go ahead and I want to go ahead and review, and I'm sorry, we're going to bring this up because I forgot to duplicate this and put it at the end. So I want to just go over this again, and we can go back over some slides if there are some questions. But I just sort of want to go through step by step what we do. We need to isolate the queen, keep our boxes covered, and establish and execute a search pattern, keeping spaces in between the frames. And train your eye. See all the workers as a blurry pattern and think of that queen as an exclamation point. And I want to make another note about restricting the light. So I'm going to go to my slide here. Okay. So I'm restricting the light on both of these. And this is the box I'm searching in that's open to the light. When bees are exposed to light, and you can do this experiment on your own. I love doing this out in the bee yard when I've got classes. What I'll do is I'll split a hive in half, and I'll put the lid back on one half and leave the other half open, and we walk away for 10 or 15 minutes. And when we come back, the hive that's been open is really, really stressed and active, and they're flying off the frame, and they're fanning homing pheromone, all kinds of things. You would think this was like some kind of, maybe it was a nasty hive or something, but then you go and you open the lid on the box that had been covered, and it's like it's a completely different hive. They're calm, they're on the comb, they're not flying, they're not moving around, and they're not stressed. Okay, so why is this important? One, for working bees, the less stressed the bees are, the less stressed the beekeeper is. And the less stressed the beekeeper is, the better job that they do. It's just that simple. But more importantly, the less stressed the bees are, the less they move. And when you have worker bees that are stressed and they're doing this, if you look at me in the video feed, they're just moving everywhere. Your eyes are drawn to motion, right? So if you've got stressed bees that are moving, your eyes are going to be more confused and less able to find that queen, right? And I'm going to go back to this video just to see if it'll let me. Come on. Where? It is not letting me. There it is. Sorry. Okay. So I want to go back to this video. And again, notice these worker bees aren't really moving all that fast and much. And that means there's less noise for our eyes to filter out. I know I use noise, that's an audio thing, but noise is a visual term as well. When there's less noise for our eyes to filter out, we can focus on that queen. So I'm going to play this 2 more times. And one of the times I want you to focus on the workers and just sort of stare at them. And then the second time, one of the times, focus on just the queen. Now it did it again. It's not good. Let me get rid of my annotation and see if that helps. There. Okay. And the workers are moving some, but they're really not. A vast majority of them are staying still, or they're not moving great distances. They're a little bit jittery. They're not covering the kind of distance the queen is. Now, the queen's probably gone 15 body lengths at this point, and some of the workers have barely moved from the spot they're in. Oops. Okay, so I'm going to play it one more time. We're going to open up for questions, and I think we're a little bit early from our scheduled time, but I think pretty close. And this frame, of course, is out in the sunlight. So it's going to be a little more active than what it was necessarily in the hive. But again, it hasn't been out in the sunlight very long. And go ahead and sometimes it's really interesting. You know, I always say when you're learning beekeeping and beekeeping skills and techniques, never take my word for it or anybody's word for it. It's always good to listen and learn, but take what I'm saying and go check it with the bee. Jamie, I'm getting a note that I'm unstable connection, so hopefully I'm not freezing up on everyone. Okay, so we're going to sort of bring it to a close, unless there's people who would like to go back and see some things. I'm happy to take questions. But again, do that experiment of opening up a hive, you know, separating it into 2 stacks and cover one of them and leave the other open. And just notice the difference. Let that go for 5 or 10 minutes. Don't do this during robbing season. Okay. But let that go for 5 or 10 minutes and then pull a frame out of the one that's exposed to light and notice how much more active and confusing visually it is. And then open the lid on that one that remained covered. And it's, I'm telling you, it's like they're not even from the same hive. That light has that much of an effect on everything. Even if you're planning to do that little pro tip about the using the queen excluders, even if you're doing a varroa mite check, especially if you've got 2 brood boxes, put that queen excluder in there, and then when you do your varroa mite check, even if you can't find the queen, as long as you've kept the boxes separated and you don't take your sample from the box with the eggs in it, your queen should be safe and secure. So even if you can't immediately start finding the queen, you really do have ways of doing those alcohol washes without putting the queen in jeopardy. And so let me just go over that one more real quick time, just for that purpose, okay? We put those queen excluders in. We don't even have to look or see where the queen is or where we think she might be, okay? And then we're gonna come back in 4 days to do our varroa alcohol wash. If, for example, this box here has eggs, we're going to set it aside and we're going to take our varroa sample from this box that doesn't have eggs, because it's still going to have a full range of brood, right? So it'll still be good for taking a varroa sample. And then that way, that just releases that fear of damaging your queen, because I get it. You don't want to kill your queen, but it's one of those things, if you don't do the mite checks, the queen may die, it may take her longer, and it'll take the whole hive with her. Okay. All right. And Jamie, I think that's about it for me. Unless there's questions, I'm just trying to scroll through there. Okay, boom. Yes, there are some questions. Linda asks, should there be a bottom board or plywood below the boxes that you've removed? Okay, so that's a great question, because it's something I didn't address at all. And what's underneath these boxes that we're stacking is very important. I have built my hive stands so that they can, a box can straddle on them and there's nothing underneath it. We never want to put a hive box on a flat surface, because we'll end up crushing bees, because there's always bees on the bottom there. But we don't have to cover the bottom for light penetration. The bottom can be open to the air. In my experience, that doesn't seem to stress the bees out. Yes, they're getting a little bit of light in there, but it's really indirect light. In fact, I usually use the overturned lid, and I put the hive on it catty corner so that there's space under there. just to stop from crushing any bees. And just see if there was any other. Oh, somebody was asking how I create the graphics for the illustrations. I play with PowerPoint a lot, and these are actually very basic. They're just shapes. I just use the create shapes to do stuff, and I've just played with this for years. It's almost like a coloring book for me. And the newer version of PowerPoint really has some good animation, smooth morphing effects and stuff. And so that could be a series of classes all on its own. All right. And I think that's it for me. I'm going to thank everyone. We got through that a little bit quicker than I thought, but we also got a pretty early start on things. I'm happy to take, good. Yes, when your computer starts telling you you're unstable, Adam is thanking me. I just keep getting these little notes, and I know the other day we were having some Wi-Fi connectivity problem here. But yes, of course, I'm thrilled to be a part of this field day, and I hope that helps with the whole theme of the field day of good, healthy bees. And healthy queens are a part of healthy bees, and especially when it comes to the incredibly important checking for mites. Find that, find that queen so that you can do the most accurate type of mite monitoring. And I've been asked to explain the difference between a mated queen and her movement versus a virgin queen. And that's a great point. Again, this is a discussion I had with some other beekeepers as well, because I've taught this class a lot, and I really do focus on your basic standard healthy egg laying queen and how to find her. But a virgin queen, so if we remember, virgin queens are preparing to take mating flights, which means they literally have to be in flight shape, flight weight and flight shape. So they are much smaller. You know, a really big egg laying queen at her peak, she's kind of like a chicken. Technically she can fly, but she may just fly enough to go off the frame and onto the ground, right? She's not going to be flying necessarily off into the wild blue yonder. A virgin queen, she's built to be able to fly a mile or more a day. And so she's much smaller. And also, the workers do something which is referred to as running the queen. So this queen, when she emerges as an adult, she's got to start mating almost immediately. Her mating really needs to be done in the first 10 to 12 days of her life. So they start preparing her for her mating flights. And so they're running her. And it looks like they're chasing her or that they're unhappy with her. I've seen some of this behavior before. And I don't think that's the case. They're just, maybe they're sort of, you know, they're like that ant. Aren't you married yet? Aren't you ready to settle down? Go, you know, keep egging and nudging on them. So they move a lot faster, and the workers around them tend to move faster too. And it's what I call runny. So when bees get runny, it's like they're really hepped up on caffeine and they're just shaking and moving a lot. And so that's kind of what a virgin does. Her abdomen is thinner, it's shorter, and I think a lot more triangular, a virgin queen. When it's so strange because, again, a queen over the course of a year, her body shape will change so much, even from the time that she emerges to the time that she starts laying her first egg. When she emerges, she should, if she was fed well as a larva, she should look almost like a fully mated queen, nice and big and healthy and fat. And then they have to get her to mating flight. So she shrinks down really, really small and, you know, ready to go fly and sort of triangular. And then when she comes back in, they start the process of fattening her up again to get her laying eggs, because eggs are expensive to lay. Okay. And the same thing kind of happens as she goes through winter. They bring her down. So the motion of a virgin queen You know, if you go in and if you think your hive has swarmed and you're trying to find that virgin queen because she hasn't started laying eggs yet, you know, that is, you've got to be really patient with yourself for any of this. Michelle has asked this question. Are virgin queens the only ones that perform pipping or piping? I was fortunate enough to witness it in a newly installed package about a week after they swarmed on me. A lot to learn in your first year. Michelle, guess what? Year 3, year 4, year 25, there's still a lot to learn. So just, that's sort of a standard baseline. But that's a great question. So piping, and Bill and Nancy Krause have a great question coming up too. Piping is, I'm not sure. I have heard mated queens make that pipping noise. Okay. And I'm not sure if there's different terms for if a mated queen or a virgin queen make it. And I've also heard and read that it's when they've fully pupated but haven't emerged from their queen cell yet, they'll make that noise as a warning that I'm coming out. So I have heard when I get a bunch of mated queens caged up and ready for sale and they're all next to each other, I've heard them pipping at each other. So I'm not sure whether it's something just for virgins that they do, or if it's something that queens do under stress or when they're around other queens. So I'm going to say a confident, I don't know, but I've heard queens make that noise in all kinds of situations. Okay. And it's really neat to hear. Okay, what is the smoking protocol from Bill and Nancy? Bill and Nancy, smoking is bad for your health, so just don't, no, I'm sorry, no. That is a great point because I totally blanked out on all of this. Here's the tricky part. A lot of times you're looking to find the queen because you want to replace her because your bees are not nice. You're trying to get a nicer genetics in there, which means smoke is your big control. And smoke does the same things to workers as it does to queens. It's the same as sunlight. It makes bees go away. So the smoke is, I say use as little smoke as possible, especially when you're looking for the queen. In fact, if you use smoke at all, don't push it into the hive. Maybe just use it around yourself, because smoke will get a queen. She can't breathe that stuff, and she's gonna move away from that. Okay, so great questions. These are, I've got to make some notes here to add those in there, because I'd never even thought about that, about smoking. I tend to, I'll tell you my own personal philosophy on using a smoker. I always have one lit and ready to go. I rarely use it. The number one thing I use it for, well, there's two things. The two top things that I use it for are when I go to put boxes back together, I blow a little bit of smoke on the top of the box to get the bees that have come to the top of the frames to go back down in, so they won't be smashed by the box I'm about to put on them. And when I'm stealing honey from the hives, when I'm harvesting honey from the hives, I will use a very liberal amount of smoke to get bees down off of those honey frames as much as possible. But if I'm doing just a basic hive, and I'm going to go ahead and do that, if that's okay. If people wanted me to bring up some of the other things, I can. Yeah, Adam says, he uses his smoker when you get stung to slow the repeats, to block that pheromone. Absolutely. Yeah, I always, it's like gloves. I go into my hives without gloves. I'm usually, in fact, I had to put a shirt on. I usually go out in a tank top. So when I got back in, I had to actually put a shirt on and I'm gloveless. And I rarely use the smoker except for, like I always have it there and ready to go. Just like I always have a pair of gloves in my backpack, because you never know, you're going to open up a hive. Maybe I'm going to be clumsy that day and I'm going to drop a box. I'm going to want my gloves with me if I've done something to upset the bees, or if my hive has requeened itself and now the genetics aren't as nice, that kind of thing. So always have the smoker there. And again, this is just my own personal philosophy. Always have the smoker there. And I use it very, very minimally, largely just to move bees out of the way so they don't get smashed by things. And to stop repeat stings. All right, any other questions? I keep getting my connection is unstable, but hopefully I'm not, freezing up, anything like that. But please, I would love, my email was up there. You guys can find me on Facebook as well, Apiary Dave, or Redbeard Bees is probably easier. But try these out. I would love to hear from you. Hey, I tried it out and it worked. I was so excited. I love getting that kind of messages. I would also like to hear from you if you've tried it out and you're struggling with this. Send me a line. And as I have time in the summer, I'm happy. I'm happy to maybe work through and see if there's something that we can tweak about your queen searching, or if there's some extra tips that I can give you. I just think being able to find the queen, it just elevates what you're able to do with your hive. And like I like to say, you go from being a reactive beekeeper, where I'm trying to keep up with the hive and react to things that have been happening, to you become a proactive manager of your hives. Because once you can find the queen, then all of a sudden you can manage the queen. And being able to find queens, I often will go out and I'll go out and I'll find 20 or 30 queens and cage them. And then that way, 2 days later, I'm ready to go out and make all the splits I want. I'm ready to go requeen whoever I want. I'm ready to go do a whole bunch of mite samples, and I don't have to find the queen at each individual hive. I break that up. I go out, I find the queens, I cage her, boom. I'm ready to do anything I want the next time. And it saves a lot. To be able to go out and just do that, finding the queens all day long, it makes the next task so much easier. Okay, a couple more questions real quick. Is it a good idea to use queen excluder between brood boxes and honey super? I'm neutral on that. I don't think it's a bad idea at all. I'm not opposed to queen excluders. I personally use them, like I said, more for managing or trapping the queen than anything else. The one time I absolutely use it for honey production is if I'm doing comb honey. Yes, Adam McDowell asked, do I mark queens normally? I absolutely do. Okay, and I think it's not only important, but beneficial in a lot of ways to have marked queens. I just suggest training yourself on looking for unmarked queens, because we're training our eyes in a pattern-seeking way. And if you're using marked queens, you run the risk of training your eye to find a dot. Okay? And so you don't want to do that. And I know not everybody can have, a row of 15 mating nooks with unmarked queens in them to check. But maybe this year, maybe next spring, you can.
Is it going to start Thursday?
Unmarked queen in a nook. You only have 5 or 6 frames to check, and you can check it over and over again before marking her. And that's a great way to get some practice. Or if you know somebody in one of the bee clubs that does have a mating yard, and they'd be willing to mentor you and come out and look at that. Usually when I teach this class, it's a half-day workshop, and we spend a good 2 hours out in my mating nooks trying to find queens. But when you break it down to an unmarked queen on 5 frames in a mating nook, that's great practice to just go through there and find her. Now, one more tip I'll give you is once you find a queen, stop, spend some time. Go ahead and watch her, just like we were watching those videos Watch how she moves. All of that viewing her is going to be imprinting into your brain what she looks like and what her pattern's like. Also, go take a video. Have your phone with you so that once you find the queen, you can take a video. And that's just a great thing to put up on your TV or your computer screen. And it's just sort of a great little refresher. Like, oh yeah, just keep watching her. And that's imprinting. It's putting that pattern, that exclamation point in your brain. Okay. Great. Well, hopefully that was helpful. And like I say, please send me some feedback and let me know as well. I love to hear success stories. Obviously, who doesn't? But if you're trying this out and you're having some challenges and struggles, send me a message and I'd be glad to talk with you a little bit more to see if we can figure out how to get you to finding queens, because it really is. It's still my favorite part of beekeeping is finding those queens, and I really love it. So thank you all very much for your time. Jamie, thank you for involving me, inviting me to be here today. And hopefully this just leads to happier, healthier hives. We can keep those mite monitoring and keep our queens safe and secure.
Number 3.
Adam McDowell.
Adam McDowell, you get 2 more hive tools. So thank you very much. All right. We're going to go ahead and let the commercial play. Everybody go out and stretch your legs, and we're going to queue up Miss Emily Mueller. And about 30 more seconds on Dave Noble's poll. So far, Dave's presentation over almost 100%. You guys absolutely loved it. It was the perfect length at 97%. And did you learn anything new from the presentation? Absolutely. So thank you, everybody, for taking the time to fill that out. And that poll, Dwayne Hathaway special. Andrew Carlin has a special on his. And then April's Greenhouse, Carlin Honey, Dave Hibbard. All right, everybody. Thank you to Dave, and thank you for answering the poll. They give us a lot of feedback, so what we know what's going on for next year, and that you guys appreciated the information, so we greatly appreciate that. And congratulations to the 2 winners. We're going to turn it over now to Emily. She does everything, the most exotic person that has done things with bees, swarms. They have their own business about doing tear outs and everything. And if it's into it, Emily's got to be the first into it, and she does extravagant things with her bees. So I'm going to turn it over to her. So thank you very much, Emily.
Thank you very much, Jamie. Just to give you a real quick information on how to contact us if you ever want to get a hold of us. He did mention we do swarm removals, and if you can't find somebody local, if you contact us, we have a lot of connections, as well as cutouts. And cutouts are kind of a specialty for beekeepers. Once they're living inside the wall, it's not an easy task just to get them out. And so we have a huge network of people who can do cutouts all around Ohio, but also a network around the entire United States. Currently, we take in about 500 calls per year at this point, not just for this area, but all across the United States. So our goal is to raise the numbers of honeybees in especially our area. But I'm very active on the Ohio Beekeepers, so this is our website. That's my poor husband that I drug along into all the things that we're doing here. And but I post a lot about swarm removals in the local areas if you're ever looking to catch bees. So with that, I'll go ahead and get this started. Forgive me because this is only my second Zoom presentation, so I hope that I do it correctly. But also I have a million children and somebody is definitely going to be trying to break down a door here soon. So if you did watch a couple weeks ago, I did a presentation on inspecting your hive. So I kind of took that presentation that I already created and turned it into more of understanding how to stay ahead of your bees, especially when you're basically spending your entire summer managing swarm prevention. And that's what I tell people is that you're, staying ahead of your bees and summer management is actually just that. You're just trying to stay ahead of your bees. And so the most important thing is understanding why you are inspecting your colonies and what it is that you're looking for, because your bees are basically an open book and you have to put the story together as you're reading each one of those frames to say what it is that's going on in your hive. So by opening up the bees and looking through all of these boxes and frames that you have, you're going to find, your story, your storyline. Is it a new queen? Is it an old queen? Is it a good producing queen? Is the hive healthy? All those things. So just real quick, I kind of want to explain what should you be looking for when you're inspecting those bees. And the main thing is your hive queen right? Because you have to have your queen and a healthy one in order for your bees to survive. It's kind of, one is the other and without the queen, the bees can't go, and without the bees, the queen can't go. So it's an equal match of health is very important for everybody involved. So when you're looking through your hive, and Dave does an excellent presentation on trying to find your queen, but a lot of people struggle with being able to, and also the nervousness that you have your first couple of years as you're going through the bees. Opening up a box of thousands of bees that can sting you can be intimidating. So if you just kind of know the basics of what to look for, you can guarantee that your queen's in there, or at least presume that she's going to be. And the big thing is eggs. So if you're looking inside the hive and you see eggs down inside the cell, you know for sure that you had a queen within the last 3 days, because it takes 3 days for the egg to hatch or emerge out into a larva. So if you see one single cell egg per cell, then most likely you have a very active queen who's laying the eggs throughout the colony. You also want to look at your brood development. Is there eggs with stages of larva? Is there advanced larva that is now pupa? Is it capped? Is it not capped? What's the pattern look like that you're looking at? Is it sporadic? Is it very close knit together? And also your sound of the bees. If you've ever opened a hive that is queenless, it's called the queenless roar. And it's pretty loud compared to what your bees typically sound like. And so when you're opening a colony and you're seeing those bees running around and they sound very noisy compared to how they used to sound, well, then look at the signs and symptoms in your hive. Do you have supersedure cells? Do you have swarm cells, or a really bad pattern going on? Because even the bad pattern can be a sign that you have varroa or mites inside, and that's going to be infecting your bees and making them ill. And it can also make your queen ill, and then it can cause other issues, viral infections and bacteria in your brood. So that's very important, is the health of your brood. What does it look like? I don't know if you guys, I'm not real great at this Zoom thing, so let me see if I can move. So this frame right up here, you guys can see that, correct? Jamie, nod, yep.
Yes,
so
ma'am.
that frame up there at the top, you can see the pattern. That's not a very good pattern. It's very sporadic, and it doesn't look great. It doesn't look healthy. It looks like some of the brood is emerging, some of the brood is not emerging. So that would say to me, hey, this brood is not healthy. The queen, something is probably wrong with her. The brood, the way that your brood looks, is going to determine the strength of your hive. If you look down towards the bottom, down here, you'll see there's holes chewed into some of these cells, and some of this looks, maybe it's dead or it's rotting or something. The coloration doesn't look right. And so as you're looking at that, I can say, oh, wow, they might actually have a bacterial infection going on in their hive, And in addition to that, mites are chewing through those cells. So what you're just looking for is does it look like there's a problem? And then process of elimination is finding out what the problem is. Hopefully most of us will have very healthy brood, and you're not going to be coming across something like that this year if you're new, or next year as a second-year beekeeper. But it's being aware of what those issues are that will keep you ahead of your bees. Let me move this real quick, because if you've ever opened a hive, and right here you see these greasy, shiny bees, if you're opening a hive and you're seeing a whole bunch of those at the top, it's very likely that your hive has been infected with varroa, and your queen has some type of viral infection, and she's laying diseased and injured bees that will not be able to produce. You'll likely see something like deformed wing virus or something in the hive as well. So if you're seeing a lot of these greasy looking bees, signs and symptoms are saying, hey, something is not right. And you'll probably also see a lot of holes being chewed through your brood as well. So, and then one of the things is staying ahead of your bees. If you're not going to add the boxes to your bees are going to decide for themselves that they don't have enough room, and they will not wait for you. They're going to go ahead and proceed with what they want to do is natural reproduction, which is swarming. So if you, I will actually show you a video clip here in a minute and understanding what is 6 of 8 frames or 8 of 10 frames, but you're looking in your box to make sure that there are bees on 8 of your 10 frames before adding a box, or 6 of your 8 frames, depending on if you're 8 or 10, to add your box, because you don't want to give them too much room. Giving them too much room can invite robbers and ants and small hive beetle and other things to come into the hive. But you also don't want to delay adding boxes, because if you delay adding boxes, then you can force the bees to want to stay in that bottom brood, and then you'll likely run into swarming issues, and the bees won't want to draw out the comb up above the box because they're already down inside and they've decided that they're going to swarm. So swarm prevention. The reason that we started our company was because we kept getting phone calls from people that said that they saw a swarm of bees and somebody had sprayed it. And so I tell people that it's, you know, is swarming that bad? No, it doesn't mean that you're the worst beekeeper in the world, and it doesn't make you a bad beekeeper. It just means typically that you're a little bit inexperienced, or you missed a massive virgin swarms after you had split a hive, which I'll explain in a minute. But the big thing is that I've had to speak on behalf of, I think it's 4 or 5 different cities now at this point that have created ordinances because people are allowing their bees to swarm in the city. And when you allow your bees to swarm in a city, then you create this big pandemic and a fear of people thinking, oh my gosh, these bees are going to harm us, they're going to attack us. And then if they unwillingly or unknowingly, spray that swarm, the hive doesn't die immediately. What happens is they become very agitated and angry, and they start flying all over the place and stinging people. So you can imagine approaching a swarm that has been sprayed and is attacking people, and you're there to tell them that honeybees are safe and gentle and kind. And had they just not done that, then it would have been okay and a beekeeper could have walked away with it. But in the long run is, had the beekeeper been managing their hive properly, the whole situation may not have happened. So one of the big things I find is that in the beginning, in the spring, when people are getting their packages and their nukes, everyone's saying, feed your bees. And a lot of the new beekeepers are feeding their bees because their equipment is not drawn out. You don't have any of the wax inside, so you need to feed those bees to get them to build up. And then all of a sudden, around June, all those beekeepers start saying, stop feeding your bees, stop feeding your bees, because if you start feeding your bees while they're completely packed inside, you force them to have no space, and then they're going to want to swarm. So you have to kind of find like the sweet spot of what is appropriate for feeding and what is not. In the early months, absolutely feed your brand new package. There is no food. But once they are completely filled into those brood chamber boxes, for most people, it's going to be 2 deeps. Once they're full in that and you're sticking a honey super on, stop feeding your bees. Because the reason for that is if you start feeding your bees when you have a honey super on, your honey is now going to be sugar water honey. And you don't want sugar water honey. You want real nectar from the plants. And so you can feed your bees up until they're filled up into those boxes, but once you're collecting your honey, you definitely don't want to feed your bees. Another thing is you don't want front feeders on your hives after May. Front feeders will entice the robbers in the area, other bees, to come over and check out your bees, find out that they have free food inside. And if you're a new beekeeper and your hive accidentally swarms, then the problem is going to be that, sorry, it's beeping and things. I didn't know what that was. So it entices all of the robbers to come into the area, and if your hive is weak, if it had recently swarmed or something as a new beekeeper, then those bees will be able to come in and rob out that hive. So just be careful about putting anything enticing in front of your colony. Don't leave any wax with honey on it, things like that, because possums, raccoons will want to come and get into your bees, possibly bears, depending on where you're at in Ohio. So try not to chill all the neighborhood bees that you have bees by enticing them with front feeders on your hives. And then I'm going to click on a video next. It's just a minute and a half long of signs of pending swarm behavior. And the biggest is there's signs before your bees swarm. And one of the most excellent books, let me grab it real quick, is The Honeybee Democracy. And this is by Seeley. And this is an excellent book to explain to you the process of swarming, and that bees actually know they're going to swarm 2 weeks prior to when they do swarm. And there are signs and symptoms that you can see inside your colony that tell you, hey, this is probably going to be happening soon. And as long as you're paying attention, you're inspecting your bees, you'll be able to find all of those signs. So real quick, I'm going to try to click on this video. Here we go. When you add a box, I don't know how to get this. Here's this. And then I started at 14 seconds in. Noticing, oh, I'm in like stage 3 swarm prep with this colony. So I just kind of wanted to explain exactly what I'm talking about and looking for when I'm telling you guys, look for these signs. The first is that they're going to start producing a lot of drone. As I'm inspecting through the colony, oh, I'm seeing a lot of drone. Okay, no big deal. Some hives just do a lot of drone. So you can't go just based off of that. But I can then pull out my brood chamber and my frames. And when I'm looking in these frames, in this area right here, you can see down in the cells is a little bit of shininess that is nectar. So the bees are bringing in nectar and they're storing it right into the brood chamber. That is not what I want because the queen doesn't have room to lay. So that is like another sign. Hey, these bees are getting ready to swarm because they don't have enough space in their hive. And then what you're going to find is now they're starting to develop these little queen cups on the bottom. This one does not have an egg, nor does the other ones that I have found also on here. So I'm still okay to work with this colony. There's another one. I'm able right now to manage the space. So they don't have eggs, so I'm still okay on my timing. I can make room, and then I can keep these bees. Now, if there were eggs down in those cells, it's a different story. You likely have to split if you're a new beekeeper because it becomes a complication if you don't. Once they decide they're going to swarm, they are going to swarm. So there are your 3 signs. Drone brood, then backfilling nectar into the brood chamber, and then your swarm cups. All right, so everyone can see that, I assume. So talking about your swarm cells and your supersedure, understanding the difference was going to tell you what's going on in your hive and if it's a good thing or a bad thing. So is it an emergency situation or is it a space issue? So the location of your swarm cells is going to explain all of that for you. Is it mid-frame cells or is it bottom? And you can see in these pictures at the top, this one isn't for sure designating where they're at. It's kind of mid-frame. So based on that, it could possibly be supersedure cells. If I saw it with the frame to the right, this one right here, well then yeah, absolutely for sure, because bees don't always follow the rules. They don't do exactly what we tell them to do or what the books say exactly. So you could have a situation where they're superseding and you have cells right here, mid-frame, or let me see if I can move this. Right here on the bottom of the frame, oops, you can see that there are cells at the bottom of your frame. Sorry, you guys, I'm trying real hard to figure this Zoom thing out here. So down here, you're having the cells They're at the bottom of the frame. I'm going to look at my drone brood, all of my brood is looking filled in, but I'm also seeing that backfilling in these cells. I'm seeing a lot of nectar. So most likely these bees had told me prior to this happening down here that they were going to swarm, but I missed all of those signs. So I missed the signs that they had a lot of drone. I missed the signs that they were backfilling. And then here you have your queen cells. But if you have a situation like this one at the top to the right, this one is telling me something is wrong with my bees. It's the brood health is not looking great. It's not a very good, strong pattern. And they're making these queen cells out of whatever was remaining. It's most likely very advanced larvae. So the queen's not going to be the best developed, but they are trying to make a queen nonetheless. So this would have most likely been a situation where something happened, the queen died for some reason, a viral infection. There's signs and symptoms saying, oh, this is probably like a varroa issue. You know, instead of you killing the queen because of what the frame looks like in general. So just to let you know that if you open up your hive and you find a whole bunch of capped cells inside of the hive, the answer is probably that your bees have already left. So if you're seeing all of the swarm cells at the very bottom of those frames, and some of them are capped, you can go ahead and dig around for your queen, but likely that they have already left because the day that they cap their cells is the day that they're going to take flight. So that's typically around day 9, sometimes day 10, and it can go a little bit later if you run into some rain or issues like that. I've heard people say, oh, I had 2 queens in my hive, and occasionally it happens. It's not very common, but weather can keep the bees from swarming if it's pretty bad outside. So what if you let your bees swarm? And I kind of touched on that. It creates panic within the city. And so I try to recommend to people, don't let your bees swarm. But the other half is that you just lost half of your bees. And so you've spent what, $500 to $800, depending on what equipment you bought and where you got your bees, on getting these hives, put them in your backyard. And if you had managed them properly, you could have stayed ahead. And if they wanted to swarm, you could split them. So I am going to touch very lightly on splitting here in a minute. But then you would have had 2 colonies. So as a beekeeper, then you say, well, I only really wanted 1 colony, so it doesn't matter. Well, you could also make your money back if you decided to split those bees, make a nuke, and then sell it. So if you only want 1 hive, go ahead and make some of your money back on selling that split. You want to really manage your hives so that they're doing what you want, which is producing honey. And I think that's kind of the goal for a lot of beekeepers, not necessarily every reason, but a lot of us do want honey. So if you're allowing your bees to fly away, you're actually the one that's losing out because you're losing your bees, you're losing the money, and you're losing your honey. And then Barb Bloetscher did talk about it earlier, and I know that I'm touching on some of, repeating some things. But the best management practices for beekeeping in Ohio is on the Ohio State Apiary website. And you can get on there and just kind of see the actual legalities of being a beekeeper. So if you are a dog owner, you have to get a rabies shot for your dog and register your dog so that people know that you have a dog and that you're a responsible owner for it. So for the same thing for bees, and I was the inspector for 3 years in Medina and Summit County. And the one thing that I ran into is a lot of people were saying, well, we don't want the invasiveness of the government coming onto our property, going through our bees. And what I tried to explain is that the government doesn't really care about your bees. What we care about as beekeepers in Ohio is the numbers. For one, we really want to increase our numbers, and 2, the health of your bees. Because if you have a colony that has American foul brood or excessive mites, and you're not doing anything to treat or help them, you're contaminating all of the hives within a 5-mile radius of your bees. And so you don't want to be the person that is creating all of these varroa issues for everybody else. And also American foul brood spreads so easily. If you're not doing something to treat or prevent spreading diseases, then you're going to actually kill the colonies within your area. Because if your hive has American foul brood and they're landing on the flowers of other colonies, then those bees are going to take that and bring that home to their hive. So I do know there was somebody not too far from my location that did have American foul brood. And what I did was I explained to them, this is the situation, this is what you should do. and you really need to get it treated. But always make sure that it's confirmed before you do something erratic like, you know, killing your hive and burning the equipment. So I do touch on that here in a second. So you have heard the big thing is varroa, making sure that your varroa numbers are low. You really, really don't want to have an excessive amount of varroa because it's going to contaminate and ruin your bees. The little cup up there at the top with this little yellow cap on it, you can get them at Blue Sky Bee Supply. I'm in Northeast Ohio, so for us that's kind of the local place, and it costs $20. And this will it'll last you years and years. It's going to be your alcohol wash cup, and you put alcohol on the bottom, you put bees into this portion, you shake it up and you dump them out, and you find out what your mite count is. $20, and you're going to be using this for the entire year for several years, as long as you don't run over things like I do with my car constantly. So, but it's going to be the best thing for you as a beekeeper is to know what your mite count is, and for the health of your bees. So something that a lot of people are talking about right now is that there is an active honey flow going on. If you have noticed, a lot of the trees are starting to end their blooms and things like that. So the honey flow is kind of coming to an end for May to June. But the biggest thing that I remember wanting to know as a new beekeeper was, well, when is the honey flow? You know, well, the honey flow doesn't follow a schedule. It doesn't start on a certain day. When you start seeing all of those blooms out there on the trees, a lot of the flowers are coming out. That's most likely when your honey flow is. And you can also see the activity level change in your honeybees. If you're looking at your colony, those bees are coming and going, you know, on a regular basis, but all of a sudden there's this huge honey flow in the area and the bees find a great source, and it will literally look like your hive might be swarming. They'll come flying out of the colony and looking for that food or going to collect the food. So, and then all of a sudden you'll notice the hive activity kind of slows down as the weeks progress, and you can, you know, pay attention by looking at the trees and looking at all of the blooms. Oh, the honey flow must be ending. So now if we have a very strong, like this year especially, we've had a very strong honey flow. A lot of new beekeepers are like, wow, this is the greatest thing in the entire world because look at all this honey. This is a very unique year, and it's been, I think, about 5 years since I've seen this much honey coming in this quickly. But then what not typically, but can happen is say, I mean, I actually have 3 mediums on a hive right now because they're not capping it. They're just bringing it in so quick. Well, in, you know, a week from now, we might get an entire 10 days of rain. And guess what? There goes all that honey. So make sure that you are paying attention to what you are taking off of your colonies so that you make sure that your bees do have enough food, and refer back to, you know, when is it appropriate to feed your bees. So staying ahead of your bee space, I will click on another video to explain exactly what I mean. But adding mediums between your deeps will allow your bees to draw out that comb when there's a honey flow going on. So say I have my 10 frames in, they're really, really packing in the nectar and honey into one frame, and they're not laying the brood inside of that chamber anymore. I'm going to take that frame out, and I can save it for another time. I can, you know, if it's not completely capped, you don't want to take the honey off of it, but the bees are going to need space. So if you can, you need to start freeing up the brood chamber area so that the bees are able to have the space that they need. And when there's a honey flow going on, you can take advantage of the honey flow by adding in empty frames and allowing those bees to build out your wax for you. Another thing is manipulating your frames. So say you have 10 frames in a box and they are drawing on everything except that, you know, the 8th frame to the right or something. Take that frame and move it in a couple so that the bees pay attention to that now the empty frame is there, and then they can start drawing that out. It gives them something to do, and it buys you a little bit of time by manipulating those frames. And I mentioned pulling the deeps of frames out, and then oh, look, I was mentioning that I was doing this presentation and this other presentation. So, but swarms, if you do have a swarm or you have questions about swarms, there are great Facebook groups. The Ohio Beekeepers page is one for the locals. There's just regular beekeeping questions groups. There's tons of groups for you to go on to. If you run into a situation where your hive, you know, you think they're going to swarm, why are they going to swarm, and how to prevent it, you can always reach out to other beekeepers online, since a lot of people are not meeting in person right now. So what is a honey flow? I kind of already discussed that. Sorry, I jumped ahead, but I just want to show you mainly. One is the black locust at the top. So that's a huge honey flow, but this year a lot of the blooms died because of the frost that we had late in the year. So last year there was tons of blooms, and then this year everyone's like, well, where are the blooms? Well, it's because of the weather. So you can think, oh, I have thousands of these trees and it's going to be a great year. Well, your frost can actually kill off some of those blooms and ruin your honey production, and your bees will have to find food somewhere else. But this is something that I ran into down here in the bottom very often when I was inspecting as an inspector. And it is people that love their bees, but they don't really manage their colonies and especially aren't staying ahead of their hive. And as you can see, when we pulled off the inner cover of that box down there, the bees had filled in all of the space they possibly could because they didn't have room, and then the colony swarmed. So what it looks like to me is that the bees were never given their second box. They didn't get to build up into that colony. So instead they filled in all the absolute space that they could, and then they left. They got out of there because the people were not managing their hive and they were not staying ahead of their bees. Had they been paying attention and watching that all this honey flow was coming in, they could have added another box, and all of that honey right there would have actually been in frames that they could have fed their bees or eventually pulled off for their own taking. So you don't want your hive to look like this at any point in time. You want to stay ahead of your hive. And if it does look like that, I do recommend scraping that off because it'll be a pain for you later on, and also a giant mess. The bees will clean it up. You can just scrape that stuff right down the bar. As the honey drips down into the colony, the bees will clean it. So don't worry about like, what do I do with it? But also don't leave it outside of the box on the ground, because if you do that, you can entice raccoons and possums and other animals to come and consume all your food. If you have not heard about the Ohio State Phenology Calendar, it is amazing. It is a website. Oh man, who did started it? I totally forget right now. I'll click on this link. But what you can do is you just come here to this website, the Ohio State Phenology Calendar. And today is June 20th, and my zip code is 44319. So I'm going to click on Show Me the Calendar, and I go down here, and this calendar is going to tell me, let's move, what is in bloom in my area right now. It also tells you of like pests and issues that plants are dealing with. Like this is the Asian longhorn beetle. So it was, the adult is emerging. So it'll also tell you other things about that are going on with your, with the blooms in your area, as well as the insects that are kind of killing all of these plants. So if I wanted, my zip code is still this, real quick, I will say, oh, okay, 5-4 works. We'll click that one. So on May 4th, if I want to go back and I want to see, well, what kind of honey am I pulling? What was in bloom at the time that I added my honey super? If I know I added my honey super on May 4th, which that's a little bit early, but then I can say, oh, well, look at all these food sources were probably within the honey, depending on what I have close to me. Now, everything that has a flower doesn't necessarily mean it's going to produce nectar. There are like catalpa trees. I think that one in particular doesn't really produce too much food sources, but some are heavy nectar, some are heavy pollen, some have zero, benefit to bees. So you could see thousands and thousands of trees with beautiful flowers, and it just does absolutely nothing for your honeybees. So if you are planting or looking to plant sources for your bees for food, make sure that it's actually going to benefit them, because if it's not going to help them with their food sources, well, then it's just a pretty tree sitting in your yard and it doesn't really benefit the pollinators. And then something that I learned recently is that bees can actually collect up to 5 gallons of honey a day. That blew my mind to think that a very strong colony that's very healthy would be able to produce and bring in that much honey in one day. So just know that, if you don't have space, that food is not going to be coming in and your bees are going to leave. But as I talk about swarm issues and all of that stuff, look at this beautiful swarm up here. It's like the craziest, it was a virgin swarm that came in. It was multiple virgins inside of the colony. When you find huge swarms like that, that's typically what's going on. But I get so many calls, like my bees swarm now. Now what do? I tried everything. I stayed ahead. I missed the signs, and now my bees left. And I didn't. Well, some people do catch them. If you're lucky enough to be within the area, you might catch them. But bees can be in your hive and gone within just a matter of minutes. And I have seen swarms just head on out, leaving colonies, and they don't come back. They're gone. So, but what do you do then if your bees have swarmed? You need to open up your colony immediately. You need to get inside and see what it is that's going on. So if you're seeing a whole bunch of swarm cells, if you're seeing 20 or 30 swarm cells, you have to knock those cells down to a limited amount of I wouldn't do more than 3, because I have seen virgins come out on just even 3 cells, but you definitely want 1 to 2 very healthy, very large, beautiful looking cells, queen cells inside of that colony. So now your bees are gone, unfortunately, but the bees that you still have will make a new queen. So go through, knock down all of the queen cells except 2 of the strongest, best looking cells. If the swarm cells are still open, here, this is what I mean. Right down here, you can see on this frame, this one at the bottom is open, and then these ones are open on the side. Well, what that actually means is that this queen developed first, and she emerged out of this cell. She chewed the hole, she chewed the bottom herself, and then she came out and she went right over here, because these cells right here were queens that were developing or about to emerge. And the reason that she most likely killed them is because they're ripped out of the side. So the queen came out of the cell, went over here, stung through all of these cells, and then they're dead queens. So the bees come over and they chew open the cells and take the dead larva of the queen out, or the dead pupa that she is now. So this is your winner chicken dinner. This queen is now going to most likely take over the colony. So when you hear about, queens fighting to the death, it doesn't necessarily mean they have to fight each other. Sometimes it can just be a matter of one emerges and goes through and kills off the other cells before she has to even fight. So, the biggest mistake I would say after your bee swarm is buying a new queen and not knowing when to buy the new queen. I'm really good friends with Laura Urban, Urban Honeybee. She's an excellent mentor to me for many years now. And she almost hates sometimes selling queens to somebody that recently went through a swarming situation. I wouldn't say hates, I shouldn't put words into her mouth, but because if you actually have a virgin queen running around in there and you don't realize it, you're just basically taking that beautiful, gorgeous queen that your breeder mated and took all this time for, and you stuck her in there to die. That's kind of what happened. Because when the bees have created their own queen and that virgin is running inside, that's their genetic line and they want to keep that queen. So if you don't realize that there's a virgin in there that's about to mate or just recently mated, but you're still not seeing eggs, they're going to kill off that beautiful $30 to $45 queen that you just stuck into your hive. And so I just kind of want to explain how to pay attention and how long you need to wait for those bees for that queen to come back and start laying. So when will you see a new queen? I already talked that the cells on day 9 are capped. Remember, that's when your bees are going to be swarming because they capped them off and your queen's ready to go and they're going to be creating a new queen. So it's going to be 6 to 7 more days after those 9 days. So day 9 plus 6 to 7, because a queen is 16 days until she's emerged. So 6 to 7 days later, that queen's going to come out after that swarm. Well, she's going to go through and she's going to kill off the other queens and all that lovely stuff is going to happen. You might even get a virgin swarm if you don't open up that colony, and they're going to leave for you as well, which will really deplete your bees. And then that whole week, she's going to spend that time probably at some point mating, depending on weather. So there's going to be that week that you just assume, this week's her sometime at one point is going to go out and mate. And so your next week after that is when you're going to start looking for eggs. So from your swarm date, it would likely be 4 weeks. If you happen to be ahead of your bees and you split them yourself, it's going to be 5 weeks. If you can stay ahead of your bees and you split your colony, then you're gaining that time for honey production. So if you split them yourself and then you put a mated queen in, you just skipped 4 weeks of having to wait for this new queen. So I do recommend, if you have to split your hive because of all the honey flow coming in, then make sure you put a new queen in there if you can afford it. If you can't, it's not going to harm anything, but don't let your bees make their own queen too late in the year, because if it's too late, then they won't be able to build up enough food and bees for winter. So, but like I said, can you add a queen? Absolutely, you can. But once there's a virgin running around in there, I would likely leave her to mate and then take her out. Give the bees 24 to 48 hours before putting a mated one in. Your virgin queen is going to be the cause of why they kill off that mated queen. You really have to make sure that you don't have her in there, because they definitely don't want the stronger pheromones of this mated queen, because they already have the virgin queen going out to mate. And then there is another way that you can prevent swarming. I just like to say, it's something that I don't really practice too often, because I don't mind splitting and having larger amounts of colonies, but you can cage your queen. So if you go in and you see all of these swarm cells, like, you know, we mentioned they're on the bottoms of the frames, knock every single cell off, every single one. You cannot have the tiniest one in production. You have to knock them all off, and then cage your queen. And you can cage her for, you know, up to a week. I would say I've heard people do it for up to 2 weeks, but that will keep your bees from being able to swarm, and sometimes it will free up some space or give you time to be able to manipulate your frames, and not lose your queen. So. But also one more tip of advice, and I talked to a few people before, is just because you didn't see your bees swarm doesn't mean they didn't. So, you know, it doesn't mean that you don't have to inspect your bees because you didn't see a swarm coming out. Staying ahead of your bees and inspecting them every 7 to 10 days through the summer for the hive honey flow is going to be, you know, keeping a good production for you. So I did say a couple things here, but I figure I'm trying to move this so I can see all of this. How to stop your swarming behavior once the bees have already decided to swarm. And if you've caught it, like the video I showed you, I caught the bees building up too many cells before they put eggs in there. If they don't have eggs in, then you can manage the frames, you can add space, you can put a honey super in between the brood chambers, especially if you have drawn out comb. You can do different things to make sure that the bees feel like they have the space. But after you have the eggs inside of those cups, there's a pheromone that has been created, and the bees know that they're going to swarm no matter what. You can't just keep on knocking the cells off and hoping for the best. They will eventually get around that or have a cell somewhere else. Once they've decided to swarm, you have to do something. So either you can continue to knock the cells off if you really want to and add space and hope for the best, but you'll have to be in that hive pretty often to make sure you're staying ahead. The easiest situation is to just split the colony. So especially for new beekeepers, instead of losing the bees or catching your own swarm, being ahead of it and splitting that colony yourself is going to make sure that you're not going to lose your bees. And I believe this next one. Okay, so splitting is an entire day of talking on its own. But I'm going to kind of just mention some successful ways to split if you are if you're a first year beekeeper and your bees have built up crazy because of this honey flow, you might have to split July, maybe even to August. So really, really manage your hive, because you don't want to split them that late in the season. So staying ahead will make sure you don't have a late summer swarm. But if you have to, these are just some tips of advice that I would like to kind of just let people know, is walkaway splits are not ideal. A lot of times people think, I think I just closed down my presentation. A lot of times people think that just taking the top box off of the bottom box and sticking a top and a bottom on it is going to save you, save your hive, and you're not going to have an issue with that. Well, if your top brood box didn't have any brood and it didn't have any eggs, well, now you've run into a problem because you split your hive, but you didn't give any resources to the bees to be able to make a new queen. Hold on, let me see if I can figure this out here. For some reason, it doesn't like me again. I don't know. All right, well, I don't know, Jamie. What am I doing wrong? It's not letting me do the presentation. I don't know. Well, you can still see it. So anyway, the common fail, like I was explaining, is that there's no eggs in that queenless box. So if you're taking the top box off of the bottom box and you're hoping for the best, if you haven't looked inside of that colony to make sure they have eggs to make a queen and you don't give them the queen, they're a dud. They're going to become a laying worker hive. So if you got lucky and the queen's in that box, well, they can produce one. But a lot of times what happens is the queen's with the brood, so she's going to stay in that box, and you think that you've done something good by splitting the hive and allowing them to make a new one. Well, the top box, they might be able to make a new one, but the other one that still has that queen, if they don't have that space, they're just going to swarm anyway. And I went out to help an older gentleman who did exactly that. He just decided to separate all of his boxes. He just took the top off, stuck a top and a bottom on it, and hoped for the best. And then all of a sudden he called me and said, I've had 12 swarms in 2 days. What did I do wrong? Well, you didn't actually look to make sure your bees had the resources, and you also didn't pull the queen out. You really have to make sure that that queen comes out of that colony so that they now, she's going to have space to lay in a new place, in a new hive. So a successful split is going to have eggs in both of your hives. It doesn't really matter how many frames you have in each. I would say a minimum of 5 in your smallest, but if you're not going to be selling it or giving it away, well, the more frames that you can divide evenly, the better. So you have 2 strong colonies instead of 1 weak one and 1 strong one. You also want to have capped brood in both hives. The weaker hive is definitely going to want to have more capped brood because they're going to be the ones that are struggling and trying to make a queen, and they need all of those foragers to go out and collect food because they've been weakened without having a queen. You need a frame of pollen. Usually frames of pollen don't come just pollen, so there's going to be nectar on it too. But you definitely need them to have that pollen so that they can feed the brood and also all of, the queen, the cells, and all of the baby bees that are going to be growing and helping create that new colony. You also want to have some honey and nectar in there. You can also just feed them depending on your situation, your setup, and how much food you have. And like I said, a minimum of 5 frames. And then when you are taking your hive colony, say your big colony, you scoot that one over to one side a little bit away and put your weaker colony where the hive was, because all of those foragers and returning bees, those they're all going to come back and help produce, make production for that colony. So you have thousands of foragers out there. Instead of allowing them to return to the strong colony, you want them to return to your weak colony so that they can help production of your hive. And then one thing is if your hive does swarm and you're waiting for that virgin queen to come back and it's been past the 21, the 2 weeks that it's been for her to go out and mate and return, and then all of the brood emerges out. Once, if you're not looking in your hive and you're not catching that your queen didn't return, you might open it up and find out that now you have a laying worker situation. So what actually creates the laying worker situation is the lack of the pheromone in your brood. It's not really the lack of your queen. It generally is the lack of the queen, but the issue is that there's no brood for the bees. And so they realize that they have to try to continue. They don't have a queen. They don't have a possibility of surviving. So they try to make their own, but they're obviously infertile. And so it becomes drone brood. The drones are a lot smaller typically than what your average size is in a laying worker hive. But you're also going to see multiple eggs per cell, and you'll see that right up here. Sometimes new queens will lay a couple extra eggs per cell when they just start laying, but it's not going to be a continued situation. It's just going to be in the beginning. So if you're seeing something like this where there are, more than 3 to 5 eggs per cell, well, something's going on. But when I'm looking at and I'm seeing those massive amounts of eggs, it's not going to just be a surprise to me when I open this hive and I'm like, oh, wow, look at all those eggs. I'm going to see other signs and symptoms too. There's going to be most likely drone brood developing. There's going to be a funky either, like missing pattern, and the bees are likely going to have that loud roar sound to them and look agitated and irritated. So how late can you fix a laying worker hive? And to be honest, the only way to do it is to add in frames of brood from a very strong colony. But to be honest, you're really not even saving that colony. You're technically just creating a new one. So you've already kind of lost the hive ones as a laying worker. You can knock them out of being laying workers by adding brood, and then they're going to try and attempt to make their own queen. So most of the time, what I've kind of noticed is that when you have a laying worker situation and then you put you put brood in there to knock them out of it, they're going to start trying to make their own queen. And when they start making their own queen after being laying workers, the chance of them accepting a mated queen is very low. That's not guaranteed, but a lot of times they are not going to want to accept a queen that's mated because, one, they were in a laying worker situation, and now, two, they're knocking themselves out of it by producing a queen, and therefore they're going to want to make their own and have that queen within their colony. So you cannot ever, I guess I shouldn't say ever, but you cannot stick a mated queen into a laying worker hive and hope that she will fix it, because she won't. The bees already think that they have a queen. Technically, they're trying to make one. And so by putting a mated queen inside of that colony, it's a death sentence. They're going to kill that queen off. So my preferred method is to just take that weak colony, stick a piece of newspaper on a strong colony, put them up on top, let the bees chew through the newspaper, go inside. They tear down a lot of the cells. They knock the bees, it kind of just tells the bees, hey, we're a queen right hive and we're not going to do that now. And in about 2 weeks, then I can take that box off and remake myself another colony. I usually will add a mated queen at that point because they've been 2 weeks in a queen right hive. So now another thing though is if you are trying to fix a hive that was a laying worker by putting brood in, watch, because they will stop tending to the massive amounts of drone brood, and you'll run into a situation sometimes where there's not enough bees because they were super weak, and so they let that drone brood rot, and it will stink very bad. It will smell like your hive is kind of dead or dying. So you'll open up the hive, you look through, and right up here you can kind of see that they're leaving some of this drone to rot and die, and that does have a very bad smell to it. So I have opened a colony and I thought, oh wow, you guys, you probably have European or American foul brood, but process of elimination found out that they actually had a laying worker. They were fixing the situation. They didn't have enough bees to take care of all of that brood, and so the bees kind of let all of the drone die off and rot out. So you can just remove the frame if your hive is really weak, take that drone brood out, that spotty, gross pattern looking, if your hive is weak, so that they can become strong and build up more. And then let's see if it's still not going to let me do, I don't know why it won't let me do a big screen.
Minimize that screen. You have two of them running.
Oh, is that why? I don't know, Jamie. Yeah, it's just one is my YouTube links.
Okay.
I don't know. See the button though? It's like invalid. Anyhow, yep. I know that Barb had mentioned the same thing that I'm mentioning on this frame a little bit. This is the 2019 inspection report, and she showed the website and where you can get this information. I know that Jamie is an inspector, and the fun part of inspecting is the paperwork, isn't it? So the paperwork is going to tell you what your inspector did, how many colonies they've inspected, what they found out. And so this is on the Department of Agriculture's website, the Apiary Division, the one that Barb Bloetscher showed you earlier today. And this just tells you how many apiaries are registered, how many hives are inspected, who had foul brood, European foul brood, chalk brood, nosema, all of those things. So it's actually very interesting to find out what do the bees look like this year in Ohio. You can compare different years. If you go back and look at all of the different inspection reports. So a few years ago, I ran into a situation where a guy contacted me and said that he had American foul brood, and so he actually burned his colony. Well, he hadn't sent in a report to Beltsville, and so when I was able to go out and assist him, we took a sample of the comb that was left, we sent that in. Turns out he actually didn't have American foul brood, but at that point he had already burned his colony. It was a really bad situation. So I I always want people to know that there are professionals out there that might tell you one thing, and it doesn't mean that, don't listen to them. It just means make sure before you do something drastic like killing your bees that you have a confirmation on something like American foul brood, because there are things that will look like it. And as an inexperienced beekeeper, you want to confirm that that's what the problem is before you just accidentally eradicate your bees instead. So, professionals do make mistakes, but always get a second opinion, because you don't want to lose your bees. If you haven't noticed, I mean, being in here all day, I've probably said something that's probably contradictory to something someone else said, because this is just the hive management that I practice, and this is what has worked for me and my bees. So when you're asking for advice from other beekeepers, do know everyone has their own experiences. So I might tell you something completely different than what someone else does. You just have to take that information, find out if it's correct or not, and then utilize those skills and apply them to your own hives. So honeybee healthy is something that you can do if your bees are sick. It is something that we have used for treating European foul brood. We drench the bees before. If you open up a colony and you say, oh, these bees are really, really sick, you have to send a sample in to find out, get that tested. Well, it takes a while for it to get tested, and you don't want to just allow the hive to die out. So what we were recommended to do is to drench the bees with honeybee healthy, and that will entice the bees to start cleaning each other. It will help them run some of that bacteria or issues that they're having out of their digestive tract, and also give them some extra resources to build up in their colony, as well as the essential oils are technically helpful for bees and overcoming things like bacterial and viral infections. So it is what we were recommended, or I was recommended as inspector to use. Honeybee Healthy will help you knock your bees out of European foul brood. It will not and cannot treat American foul brood. As far as I know, the only method is ozone, and I don't know anything about it, so I cannot help you with that. And then Barb also mentioned poisoned bees and reporting that. My second year, I ran into a situation where 18 of my colonies died over a matter of 2 weeks because my bees were contaminated. A retention pond was sprayed with some type of contaminant. My bees brought it back, and it killed off my entire apiary. It was a very horrible year. And so what I found out eventually was that I was supposed to report it to EPA. I was supposed to report it to the county inspector and also the pesticide division. Your county inspector or the state apiarist will be able to assist you with finding out how to do it, but it is literally a matter of get it done immediately. Because of the half-life of the products that are used, they might not be able to see the chemicals that killed your hive or your colony. So just know that if you run into a situation where you feel that your bees have been poisoned or a pesticide issue has happened, immediately contact your county or your pesticide division, your county inspector or the pesticide division, so that they can come out and get a sample of it and make sure that people know that those things are harming your bees. So, all right, with all of that said, if anybody has any questions, feel free to ask away. I don't mind answering anything. So
I have a few for you.
All right.
It says, if you have new undrawn comb frames, would you recommend 1-to-1 sugar water to entice the bees to draw more comb?
Well, it really depends on, are they in your brood chamber? Where are the frames at? Yeah, you can feed your bees, but make sure that you're not feeding your bees if you have honey supers on, because you don't want that sugar water to go into it. I've heard people, I personally will take a hive and say they're not building on it correctly. And when I'm pulling that frame out, they'll kind of build wonky comb, and a lot of times it has nectar. I will take like that brood comb off and I rub it onto the frame that they're not building off of. And the wax and that nectar usually stimulate the bees to want to build off of it. If your frame doesn't have a lot of wax, they're not usually encouraged to do that. So you can just take burr comb and rub that on, or you can actually melt down wax and apply it with a paintbrush. But yeah, sugar water, I mean, I've heard that it can help. I don't personally know spraying it with sugar water is going to get them to draw it out.
All right. Also, the same question from John. If you have a queen cup, do you recommend pinching it out?
So if I'm going through my hive and I'm just seeing a couple queen cups and there's no eggs in them, no big deal. Don't worry about it. The bees are going to have backup cells. Real quick, let me actually show you. I forgot to mention that. Is it this frame? Where's the, right here. So is it this one? Let's see. Move. It's not. So there was one where there was a cell at the bottom of a, here we go, right here. Okay. So when I'm looking through my hive, you're going to, no matter what, you're going to most likely find some cups, and there's no development on the cups. And if you can see down here, I'm showing you these queen cells. This one's a really strong, healthy queen cell. This one is, this one's growing. And then you got this little guy right here. Well, this little guy probably doesn't have an egg in it because the development, I'm showing that they, you know, they're developing these, they're not developing this one. So after these queens emerge and whatever happens is going to happen, this one will likely just hang out here, and it will never, they'll never tear it down because there's nothing inside of it. So this one is probably just going to be there on the frame, and the only person that it bothers is me because I get paranoid that maybe they're going to put an egg in it. But if I don't have any other signs and symptoms in my colony that are saying that they're going to swarm, it's just a backup cell that they have hanging out for no reason.
And if anybody has any other questions for Emily, please type them in the group chat.
Let's see if I know how to get to the group chat. I can show you this other video real quick too, if you have a second. Well, but this is just explaining when to add a box. Maybe.
And I tell them when they're on 80% of their frames to add another box. So I checked this hive last week, and they didn't build out very much, but with the incoming honey flow that we have going on, they have definitely packed in into some of the frames that they weren't on last week. So I just want to show you, this is what I mean when they're on 80% of your frames. I can see bees on this frame, all these frames, and they kind of stop about right here. So these two frames are still being built on, and I'm going to pull out this frame down here to see as well, but they're not on the outside edge of this. So they are officially on about 75 to 80%. And so now at this time, I'm going to add a box. This is actually a little overfull. I could have probably added a box a couple days ago because of how extensive this one is packed towards the edge. So it would have been okay for me to add a box had they kind of been more towards this frame instead of the entire box the way that they are. So.
All right. Any more questions?
Just one. How can members get added to the Tear Out Association?
You mean to Mueller Honeybee, like get on my list for cutouts, that kind of thing?
That's what I'm guessing from this.
You can just email me on my website, or if you type in Mueller Honeybee, my website or information pops up. My number on there is also my cell phone number, so you can text to that number, and I usually just save you in my phone. When you do text me, tell me what you do, if you do cutouts or you do swarms or trees or all of the above, and also what area you serve. If you say all of Ohio, I'm going to ask you what area do you serve, because I try to keep the people within their areas. You know, I mean, it's great that some people will travel 2 to 3 hours, but let's let the beekeepers down in those areas get those hives so that they can also build up their experience as well.
Personal question from me. Do you have anybody else up in this area?
Northeast Ohio, because I think you're on
Northwest.
Northwest. Yeah, there's a new guy, Dylan, I think.
Okay.
There are a few people out there. I probably have them on my phone.
Okay.
So,
it's just us older guys are getting tired of doing tear outs.
I feel ya. I feel ya. I'm not, I mean, like, I'm older now, but I'm still kind of tired of doing cutouts. Like, yeah, it's not fun. There's nothing fun about it. And people will say, you know, like, you should not charge because it's free bees. Well, I'll tell you what, spend 10 hours getting stung in a bee suit in a 100-degree attic and let me know how much fun that is for you. So.
All right. Question is, what is the best month to start a hive?
The best month, I mean, it depends on if you're doing a package or a nuke. If you're doing a package, you typically get them in April or in May. If you're doing a nuke, you usually get them May, June, July, and I even sell nukes all the way into August. So just during the beekeeping season, you really want probably 2 to 3 months to be able to build up your colony. So I tell people who buy, you know, August nukes, feed them until they're completely and totally packed full. Obviously stay ahead to make sure that they aren't over full in there, but you can buy hives in between April to August typically and be okay.
All right. I'm going to launch a poll for Miss Emily.
So what happens if they
everybody will
don't
take
like
a few
my
minutes?
presentation? Do you tell me like you only got 30%?
I know. I do not.
I'd like
I would share it either way. I'd share it either way. But everybody's, they really like the education side of this. You had excellent pictures, Emily, and a lot of the things that I talk about is the same things. And it is on each independent beekeeper, the micro environment and everything that affects different beekeepers. So like she said, get good educated. She had great things from ODA, Honeybee Health Coalition, things like that. And ask those questions. Just don't go out on YouTube and just get general questions from people or a second-year beekeeper that all of a sudden started something up and he's trying to answer questions.
Yeah.
So excellent, excellent. Thank you very much, Emily.
You're very welcome.
And if there's any other questions, please type it into the group chat. And if not, we'll start wrapping up our day.
You made it. You survived.
A lot of great feedback throughout the day. So, Miss Emily, from so far, we're going to keep the poll open for about another minute. How did you enjoy Emily's presentation? Everybody said yes, absolutely. The length of your presentation was perfect, 97%. And did you learn anything from this? 100%.
Cool beans.
So, did a great job. Thank you very much.
Thank you.
And we'll turn this over. I'm sorry, go ahead.
How many participants were allowed? I was curious. Is it 50 or 100?
I have a license for 500.
Oh, cool. Sounds good.
And we average right around 57, 58. There's 60 or 46 on the actual chat, and then I have about 18 that it's been fluctuating that have just been listening to the phone call that I have patched in through my mixing board.
Cool. You had a really good turnout. Thanks so much, Jamie.
You're welcome. Thank you. And for everybody else, I'm going to go ahead and run the specials real quick for one last time. If you guys are interested in anything, remember Dwayne Hathaway has 10% off his equipment from yesterday's date all the way up to July 4th. He has a special on honey bears. We had April's greenhouse. We have Carlin Honey, 15% off his Etsy page with a promo code. Myers Honey, which Roger does tear out as far as I know. And we have Brad Hibbard that has the Honey Blossom Orchard, which is also a supplier in our Northwest region. And then he also has the thermal industry, the thermal pads. So we'll come down to the last one. I want to thank everybody for the sponsors and everybody that participated and spent your afternoon with us. Thank you for the presidents that stayed on the line, and especially thank you for all the presenters. Everything went real smooth. Thank you for your cooperation and sharing all your great information with us. So I greatly appreciate that. So for our final, number 29. If somebody can find that maybe quicker than I. Miss Dorothy Brown from Northwest Ohio Beekeepers. Are you on the line? Dorothy Brown is still on. So Miss Dorothy Brown, you have a gift certificate for $50 for Dwayne Hathaway. I'll get that to you through Dwight Wilson or myself. Thank you, everybody, for your time, your patience. And we had a great time today. Everything went really smooth. Yep, and Dorothy said she's there. So thank you very much, Dorothy. So I'll let the commercials run if you haven't seen them. I'll unmute if you want to unmute yourself and if you have any questions. I do want to thank Adam and Chris for keeping track of and signing the numbers with everybody on that. Any final words, Mr. Roger? There you go, Roger.
Yeah, now I can talk.
My fault.
It wouldn't let me. That's why I was waving. Yeah, I want to thank everybody also. I want to thank Emily if she's still here and all the other speakers. You know, without you guys, our clubs listen to Jamie and I blow all the time, so it's good to have somebody else talk. Yeah, I had to switch shirts before I got here today. I already went through 15 hives this morning, so I got my hives in early. Chill out the rest of the day. Thanks everybody for showing up. You know, Jamie and I and Russ and Dwight, you know, we really enjoy helping you guys and teaching you to be better beekeepers. We have other things to do today. So, you know, if nobody showed up and it was just us four here, we'd get tired of looking at each other. So, thanks everybody for being here. We appreciate it. And maybe next year we'll be able to do this live and in person again and get that good potluck food that I'm really disappointed about. I had to eat a roast beef sandwich for lunch today instead of having some good potluck. So, thank you, everyone.
Mr. Russell Bowes.
Yeah, so I just want to thank everyone too. The presenters were very good. I really appreciated hearing their presentations. And Jamie, thanks again once again for putting this all together and hosting it. Really appreciate you hosting also our club's Zoom meetings. I appreciate that.
Not a problem. And Mr. Wilson, the wisest to the last.
All right, there we go. Jamie, thank you for putting this all together. I think Roger and Russ said it. You know, we want to thank you and the participants on the day. It was just wonderful, a lot of education, and I think people learned a lot. I certainly did. So with that, now we can go out and be better beekeepers. So I look forward to the next time we all get together. So with that, have a great evening.
Have a great evening, everybody.
Thanks, Jamie.
Thank you. Have a good day, everyone.
Thank you, Jamie.